If everyone could sit down, we can try and keep on time today for our second day. All right, welcome everyone and good morning for GMI Day 2. I was very proud of everybody yesterday. We sort of kept to time. We had lots of good questions in the panels and yeah, plenty of food. Sorry about the toilet situation for the women. Hopefully that should be fixed today. Today's a bit of a different day. Yesterday we pushed you hard. We went from 9 till 7 for those of you that stayed to drinks. Today's going to be a bit simpler. We had the great early career network this morning. I thank Masami and Adrian and others for organising that and actually attending, and it was great. It was a good turnout. It was better than I thought. I was actually excited to see so many people there. But today's a bit different. First we've got a session on data sharing, which Adrian is going to chair in a second, and then we've got a workshop that Tim Dorman is going to run. And then we are going to have a lunch, but it's not going to be here. It's going to be off-site, and Tuyet will give us details about that later on. Yeah, and there will be animals. Now, you may be thinking the weather is a problem. Now, I promised you yesterday it was going to be sunny today, and I rang up my friends at the Bureau of Meteorology, and they're going to do what they can. I think by 2 o'clock it should be sunny and the rain will be minimal. So it looks a bit terrible now, but the sun's up there evaporating the clouds, and I think we should be good for this afternoon. But it is my pleasure now to introduce Adrian Ingle again. He's going to chair the session. Unfortunately, the original chair, Francis, couldn't make it. The classic visa delays and problems like that. So it's very unfortunate that he couldn't be here, but Adrian stepped up enthusiastically to take over. So I've handed over to you, Adrian. Thank you.
Thanks a lot. Thanks a lot. I think there's a discrepancy between the data and the expectation with the weather. So we will see if your prediction model is right. So, okay. Session 3 is all about data sharing, and we had yesterday already the opportunity during the panel discussion to, emphasize the importance of data sharing. So in this session, we now focus on data sharing and integration, and I'm really excited to also see now speakers from low and middle income countries across Africa, Asian Pacific regions, and we will address a series of key questions. So for example, what can we do as a community to identify, address, and overcome some of the challenges and risks of data sharing? As Torsten has already mentioned, our first speaker is not available, unfortunately. So we start with our first speaker, Yuping Hong. Yuping Hong is a senior clinical microbiologist at the Taiwan Center for Disease Control, Ministry of Health and Welfare, and she leads the national incentive to implement whole genome sequencing-based strategies for pathogen monitoring and outbreak investigation. And she will tell us about her perspective. So, please, you can come on stage. Thank you.
Hey, hello everyone. I'm Yuping from Taiwan CDC. Really nice to meet you all. Yeah. Today I want to share my opinion of data sharing and how it supports our surveillance work in Taiwan. Yeah. Yeah. Okay, this is where our country is. It's very near China. Yeah. Okay. To begin, I want to introduce a journey of the genomic surveillance of enteric pathogens in Taiwan. And in 2004, we joined the PulseNet International. We are adopting the standard PFGE for Salmonella surveillance. Yeah. And in 2015, we introduced WGS technique. For the research and the pilot program. Yeah, we use it for outbreak detection and some AMR monitoring. Okay. In 2022, we joined the PulseNet Asia Pacific training facility study, and our team completed the Illumina training. Yeah, and we implemented harmonized SOPs and QC metrics for the regional data sharing. Okay. And by this year, we fully transitioned to the WGS-based national surveillance system. Yeah, we use it for a more quickly action and for the global alignment. Okay. This is the national framework for enteric pathogenic surveillance. Yeah, the pathogens are divided into passive surveillance, such as Salmonella typhi and Paratyphi, and the Shigella and the Listeria monocytogenes, and Vibrio cholera. And another part is about the active surveillance. Yeah, it's project-based, such as some non-typhoidal Salmonella and the Campylobacter and the GBS. Yeah, we collect all the isolate from hospitals. And when we receive the isolate, we conduct whole genome sequencing and AST. Yeah, and from the WGS data, we can predict the cell types and AMR genes. And we also use the whole genome sequence data to do cluster detection and doing the global alignment on NCBI database. Yeah. Okay. This is an interesting case. Yeah. This is the first imported XDR typhi case in Taiwan. It was detected in 2019. In 2019, yeah. The man is a businessman, and he traveled to several countries. You can see the travel history, yeah. When he returned to Taiwan, he had persistent fever and diarrhea. Yet we want to know where was he infected with the XDR typhi. So we do the whole genome sequencing and use the global alignment on NCBI database. Oh. Finally, we combine his epidemiology information and the genomic data. Oh. Fortunately, we finally identify the most likely place of infection. Yeah, it's Parkinson. Yeah, okay. And also this year we detect a new XDR typhi case, and our direct asks us whether there is local transmission in Taiwan. Yeah. To answer this question, we compare the 3 isolates we collect these 3 years, years from 2019 to 2025. Yeah, and according to this final trajectory, yeah, we find they are differed by 12 core genes different. Yeah, so so we think it is most likely from different source, not a single transmission chain. Yeah. Okay, next. Okay, this is an overview of newly emerging MDR and SDR Salmonella in Taiwan. Okay, in 2015, we detected MDR antigen. It has 11 AMR genes, and it's all on the plasmid. And in 2018, we detected a serotype Gold Coast. It harbors a large plasmid. It owns 15 AMR genes. And then in 2021, we find a chromosomal-borne variant of infantis. And then in the 2 years, 2023 to 2024, we find the mega plasmid, which was found in Gold Coast, was transferred to Agona. And we also detect a chromosome-borne variant. Yeah, and last year I have also reported erythromycin-resistant XDR Salmonella typhimurium. Yeah. Okay. Oops. Okay. And these findings we these findings are published in the surveillance report or journals to promote awareness. Okay, this slide summarizes the genomic data we have uploaded to the NCBI database. So far, we have deposited over 5,000 entries, and it covers Salmonella and Shigella and Campylobacter, Listeria, monocytogenes, and Vibrio cholera. This is the conclusion. Okay, data sharing enables real-time disease surveillance. By sharing data, we can achieve early warning and better control of antimicrobial resistant trends. Yeah, and second, only accurate and well-curated data can support reliable public health actions. High-quality data are foundation for sound epidemiological interpretation and decision-making. And the final is, I think it's the most important. We should respect and value the data contributed by others. Generating data requires effort. We should share this data responsibility and appreciatively. Okay. This is my colleague. Thanks for their effort. If you are interested in these MDR trends, you can refer to these articles. Thank you.
No time for questions?
No, there is time for questions, actually, because we are a bit ahead. Please, go ahead. We have a panel session afterwards, but perhaps a short question.
Quick question, quick. Go back to the front. So, we're great to see you sharing your data, but there are a lot of people in the room that don't want to share their data. So I'm curious, what was the barrier that you got around? You know, what was the most difficult part of convincing your folks to share data?
I'm a representative of our government, so I want to say that our government likes to share data. The policy is open, yeah, so we should open data. But also we are afraid that the data was misleading or for the abuse usage. Yeah, I think it's the difficulty of others on sharing data.
Okay, thank you very much. So great, we come to the next speaker. This is a local, Danielle Ingle. Where is she? Ah, there you are. Very good. So Danielle is a NHMRC fellow and is based here at the University of Melbourne. Her research program is centered upon high priority bacterial pathogens, including serovars of Salmonella enterica, pathotypes of Escherichia coli, and Shigella species. So please, the stage is yours.
Cool. Great. Cool. I did the classic thing where I tweaked my slides based off yesterday. So I will keep to time, but I will just be keeping an eye on my phone. So today I'm going to be talking about, obviously, going from how we can use genomic data to making it inform public health action. And today I'm really going to be focusing on the far side of the knowledge framework, so how we're interrogating these genomic data, the analytical analytical platforms to ensure that we're using the same nomenclature, which again was highlighted yesterday, and also how we can make it more accessible to those who aren't in the genomic space or research space. And the pathogen that I'm going to be using as the exemplar is Salmonella typhi. So a bit different to some of the talks from yesterday, which were more country focused. Today I'll be really focusing on the work with the Global Typhi Genomics Consortium of how we're working together with a view of better exploring and disseminating information about this bug. So typhi was one of the earliest sequenced pathogen genomes just over 20 years ago. So there's been a lot of time in this space to work out, what we want to know. There's a pretty established nomenclature with how we talk about typhi, and that's shown on the far side with the genotyping tree and the different lineages. We've established, the AMR of importance. Typhi has very specific definitions for what is an MDR typhi, what is an XDR typhi, and the underlying genetic mechanisms. And we also have a clear understanding of the plasmids in particular that we care about from an epidemiological perspective. And in the previous talk, we had some of the Inc. H1 circulating in some of their strains. And so what the Global Typhi Genomics Consortium sought to do, that was established, I think in 2020, was that it seeks to bring together everyone working in the typhi research space and also public health, and it has three broad overarching goals. Obviously, sharing of data in a prompt fashion, trying to extract and report the key data in a way that's readily accessible, and then promoting the use of this information to inform public health and potentially vaccine strategies. Oh, that didn't display properly. So what the Typhi Genomics Consortium initially sought to do was back in 2020 reach out to everyone that was working, who'd published on Typhi, and invite them to join the consortium. Nearly everyone signed up. And what this meant was that there was a total of 80 different data sets at the time. To the question earlier, 20 of these were unpublished. And this was underpinned by everyone who signs up to the consortium has to sign a memorandum of understanding. And this is really to embed that trust if you're going to share that data, so we can do analysis before you publish, that we're not going to do the dreaded scoop you. And that is really pivotal to the consortium's work. We did have a metadata. There was some discussion around this yesterday. One of the things for typhi in particular we cared about was capturing both purpose of sampling, so there was some discussion there, but also country of travel, particularly if it was from typhi sequenced in a high-income country where typhi is not considered endemic. Capturing that reported travel data and using that to infer country of origin is really important. And so this was one of the first outputs. It was a publication. I'm not actually going to talk about any of the results, but just to highlight, this first publication was really to provide a meta-analysis of nearly 13,000 genomes that enabled us to make some insights into the emerging AMR profiles globally, and also be able to drill down into countries in detail where we had the sampling was sufficient enough over a 10-year period to make more insightful interpretations of emerging AMR. But given the discussion, I also wanted to talk about data sharing in how we're working in the specific working groups. So that first publication was one working group. There are currently 4 active working groups. I'm lead of 2, the phylogenomic and the typhi microbe. These working groups are open to everyone in the consortium. And so initially there's an expression of interest email circulated. And then we use Google Forms to capture the details. Importantly, at this time, each of the projects has a really clear objective with a clear statement of what we're seeking, as well as clear expectations of what warrants authorship. And I think this is, again, important if you're trying to build momentum, being upfront so no one turns around and says, but I did the thing, and you're like, so we're having a clear expectation there. So drilling down into the typhi microbe working group, because I've been working on this one more recently, this is an example of what we're doing in this space. So this working group has over 30 members, all continents, obviously not Antarctica. I think with the data sharing, often there's a lot of how we share the genomic data, how we share the metadata. Really importantly in the working groups is not only the initial sharing of genomic data if it's unpublished, but sharing of ongoing analysis, ongoing interpretation, and ongoing how we're going to try and publish these results. A really important thing is obviously time zone considerations. I will flag that if I'm leading a meeting, there are certain hours of a day that I just don't function from midnight to 6 a.m. But that means that doesn't work with a lot of the rest of the world. So because we're really aware that everyone's in different time zones, it means that not only when we're having the working group meeting, there are communications sent both before with a clear agenda of what we're going to talk about. Slides are then sent after that I've presented, as well as key action items that we're going to work through. And this is so that if you can't make the meeting, there's not a sense of, I can't, just because I can't make one meeting, I can't be a part of it. It's trying to ensure that everyone has that opportunity to engage and to share in a way that means everyone has access. The other part as well is all of the underlying code and the analyses, as well as when we're working on the drafts, are all open access to all the consortium members. And again, I think that's really important in building that momentum to share. A good example of the Memorandum of Understanding specifically was for this Typhi microbe project, we wanted to get matched Illumina and ONT data. And we wanted to have this for a specific part of this validation. And Aruka's group in Nigeria was working on their own paper for which they generated 3 genomes. And because we had this Memorandum of Understanding in place, they were happy to send me the data 12 months before they pre-printed. And sorry. I knew I was rambling. Okay. So we've got 2 analytical frameworks, one of which is Typhi microbe, direct from reads, and it enables us to get to that key information that we like to look at in a Typhi genome. Pathogen watch is the other one. We're also able to then link this to where MIC data has been made publicly available, in this case the CDC and the UKHSA, and show categorical agreement for the genomic determinants and AMR phenotype. So this is where we are with these key digital resources that underpin Typhi genomics, our metadata, our standard nomenclature, and also this AMR dictionary. And these are implemented into Pathogen Watch and Typhi microbe. But the final part is then taking it to a point where people can actually explore it if they aren't researchers or bioinformaticians. And so this is TyphiNet. And this enables you to look at all the publicly available Typhi genomes. It pulls the data from Pathogen Watch, one of the frameworks that uses these schemes that we've implemented, and enables you in this case to identify, in the red, we have Pakistan where XDR is emerging. You can drill down into each country. So for instance, you can see on the graph closest to me that XDR emerged in about 2016, that it was mediated by 2 specific determinants that are associated with the lineage in black. And we can see that that lineage has really taken over and is now dominating all of the other Typhi lineages in that country. And so with that, I just wanted to highlight the digital resources that we're building in the Typhi Genomics Consortium space, both the resources, but then also taking it that next step of how we can curate the data and make it accessible to public health and epidemiologists so that they can hopefully inform policy and public health surveillance. And so with that, thank you. Sorry about going over.
Thank you so much. I think the bing sound is when there are 3 minutes left. So yes, you had some time. But please, I would like to invite the people from the side to actually come close, a bit more in the center if you want. There are still several seats here and here. Okay, so we come to our next speaker. This is Ridhuan Ali. I hope I pronounced correctly your first name. He is a microbiologist at the Institute of Medical Research at the Ministry of Health in Malaysia and holds a PhD in medical microbiology. And he contributes to the national efforts in genome surveillance and especially whole genome sequencing for AMR and emerging pathogens. And he will share with us the foodborne disease surveillance perspective. So please, Ridhuan. Ah, here you are. Very good.
Thank you, Prof. Adrian. Hello, everyone. My name is Ridhuan. When you do Illumina sequencing, you will remember me. You're Ridhuan. This is the title of my presentation, and above of me, this project also is part of a group works by Jama in the middle, in green color, and Hazrin in yellow. Okay, this is Malaysia to put some things in context. And Institute for Medical Research, we in charge, belong to the Ministry of Health Malaysia. We in charge of more than 140 hospitals and more than 3,000 clinics. A part of that, we also have public hospitals, army hospitals, about 13 of them. And MOH also govern more than 200 private hospitals and private clinics. So in terms of samples, we have a lot. And we were established in 1900, about the same age of London School of Hygiene and Tropical. And we have 8 research centers, and Infectious Disease Research Center is where we belong to. And under Infectious Disease Research Center, we have bacteriology, virology, archaeology, entomology, and parasitology units. And mycology units is covered by our unit. And because infectious disease is governed by a special act in Malaysia since 1988, it is mandatorily notifiable disease to the MOH. This is the data that I collect, I check between before COVID and after COVID. We see a trend, a decrease in trends. But post pandemic, the numbers going up to about 50 incidents per 100,000 populations. In terms of numbers, I think we are back to the incidence of food poisoning back to what we had before COVID, around 16 to 17,000. And then of this, about 117 are due to typhoid and paratyphoid, 514 for dysentery and cholera 47. We had quite low number of cholera cases in Malaysia, and it is very restricted to a particular region neighboring Philippines. Once we were invited and to PNI, we let our hospital know that we can now sequence enteric bacteria, and that is what we have received so far, and it is increasing. And then from here, for close to 500 isolates, we have sequenced 178 using Illumina and more than 100 using ONT. And if you can see, a part of enteric bacteria will also start sequencing Neisseria, Elizabeth Kingia, Streptococcus, and many more, including fungal. Because once we have the reagents in our lab, we start having clinicians, our clinical microbiologists in the lab, asking whether they can slot in 1 or 2 samples from their clinical cases. And then back to the main question, can NGS data and its metadata from MOH be shared? To answer this, I look from the literature, and then one paper came out from Emeritus Professor Sheikh Junin, a Malaysian researcher. This was an old paper and still relevant today, looking onto what are the challenges, what are the current practice lies between data sharing in Malaysia and few other Asian countries. And also I checked the NCBI. There are a few sequences. We can see names, IMR names on the lower part. So I think we are good. At least some of sequence are already there, at least for Salmonella. And then we had a few for Aeromonas, and only one for Vibrio cholera. And then when we did homework, at least these are the acts related to data sharing. And recently, in April, the Parliament passed another bill, so-called Data Sharing Act. And then from MOH perspective, we also have our own policies and regulation. And yes, it is based on these 2 and other regulations, and also some international standards. Glad that IPSN also put these slides on. And then at the end of the day, I think we want to really take care of what we have in Malaysia and those who live in it. When it comes to data sharing in Malaysia, I would like to put it back. Based on this data, we know that MOH potentially is the largest numbers, manage the largest numbers of hospitals and clinics in Malaysia. So we are potentially the largest health data generator. And there are many data collected, and I can assure you that some of them use ICD-11 to ensure standardization. And a part of that, we also have more than 15 disease patient registry, cancer registry, patient registry for particular needs. And because of this, we have really clear policies and guidelines on how to establish a registry, and these registry are accessible to other researchers. From here, you can learn how you can access the data. Typically, you need to write to the MOH, and then you can access the data. And this data is governed by our sister institute, so-called National Institute of Health in Setia Alam. And a part of that, many of MOH data is also governed by health informatics centers. This is located in Putrajaya. And a lot of data also are contributed by diagnostic lab research and surveillance. And IMR, even though in our name it is called Institute for Medical Research, we really played these three roles: diagnostic research and surveillance. And when it comes to how IMR do data sharing, there are a few options. I think patient registry is too big for us, but we can use the model to do data sharing. A part of that, our sister institute also create this data repository system. This is quite new, unlike the other countries that have very well-structured metadata, but it is a really beginning for us. And in our team, we prefer to use publication through journal articles and public database. It is very conventional, but this is what we have currently because we have very clear guidelines on disseminating this kind of data. Okay, in order to enable us to share our data, for PulseNet International study, we registered this study, and then we obtained ethics approval. And then with this ethics approval, we plan to publish everything, but this is the priority that we want to publish. The first case will be Campylobacter fetus, species tested. It was our first time seeing this organism when we asked the hospital to send Campylobacter, and then suddenly they sent these two fetus. It was our first time, and we found out that these two patients live about 100 kilometers from each other. This is very rare. We were so worried if this is a potentially outbreak cases. One of the patients died, and then we did some SNP analysis, and fortunately it is not an outbreak. And we were invited to, this is National Pathology Conferences, of which all pathologists, clinical microbiologists of Malaysia governed in one state, and then had a meeting. And then over there, we were invited to publish, to present something on whole genome sequencing. And I would like to thank these people. Thank you very much.
Thank you very much for this wonderful talk, and it's also great to see that you have such a strong legal basis also, and yeah, it's very well regulated. So Campylobacter fetus is indeed a fascinating pathogen. We also had recently 2 people who had a sepsis, and there was a question if this was related. Yeah. Cool, thank you. Okay, then we come to the next speaker, because afterwards we have a panel discussion. So the next speaker is Ebenezer Foster-Nyarko. Eben is a postdoctoral research fellow at the London School of Hygiene and Tropical Medicine in the UK, and is the co-coordinator of PulseNet Africa. So please, Eben. Oh, there you are. Very good.
Good morning. Shall I share from my laptop?
Yeah, need to press next because I have many slides.
Okay.
You press the green arrow and then
Yes.
it moves forward.
Which one is the green
green.
arrow again?
Big green arrow on the
The big green arrow.
Yeah.
This one.
Yeah, there's a very big green
Yes. Right. Thank you. So my name is Ebenezer, and I'm a postdoc in Professor Cathold's lab in London School of Hygiene and Tropical Medicine, and I also serve as the PulseNet Africa co-chair. I'm privileged to be here. Thanks to the organizers for inviting me. So PulseNet Africa is like a regional collaboration of public health labs across Africa, which I've shown in the plot on the right, the current participating countries. So we've been tracking food and waterborne diseases since inception in 2010. And the pathogens that are of concern to us are Vibrio cholera, Campylobacter jejuni, Salmonella enterica, enterotoxigenic E. coli, and Listeria, the key players across the world. And we sit within PulseNet International, which I've shown to your left, which brings together labs from more than 100 countries in 8 regions. And the beauty of PulseNet International is that we all work into the same protocols and using the same kind of nomenclature and so on. And so that's really helpful to compare methods and work to the same standards. From 1996 until July 2015, we utilized pulse field gel electrophoresis to determine and identify clusters. But since 2019, we've transitioned to whole genome sequencing. The integration has been different across different regions. So when it was pulse field gel electrophoresis, it was easy. When a site identified some pulsotypes, they would email that across to Anthony in NICD, and we'll compare to existing classes to determine if there's relatedness, etc. But well, let me step back a little bit. So this is the model. So in Africa, we've got like 91 million people falling ill from foodborne diseases every year. A subset of these will visit the clinic, and then a lab technician will identify the key pathogens of interest. This will then be sent to the national public health lab. There will be sequencing, and then, as I said, the data would then be transmitted, and gels will be compared, etc., to see if there are classes, and then epidemiologists would investigate that. It was super easy to transmit, gels, etc., by email, but during the COVID pandemic, we saw an explosion of capacity across Africa. Africa PGI capacitated a lot of labs, and so we now have so many labs, having been able to do sequencing. We've got a lot of centers of excellence, down from 7 at the start of the pandemic to about 30 now, operating as centers of excellence, and you can see capacity has increased. And with this comes the problem of big data, which is quite significant. So if you look at the table, with estimations of about 15 terabytes of data generated weekly, in 5 years, if we operate at half the capacity, we expect at least 1.8 petabytes of data, which is quite significant. And we also are interested to share the data locally within labs in the country, regionally, and then across Africa. And so data governance is something that needed to be discussed. So there was a creation by Africa PGI of the Pan-African Genomic Data Platform, which began to discuss, platforms for data sharing, architecture, governance, etc. A number of working groups were set up. For example, there was a foodborne disease technical working group, which I was privileged to chair. And one of our outputs was to come up with a roadmap and implementation strategy for how genomic surveillance of foodborne pathogens was to be done across the continent. And on the right, I've shown a plot, a figure from our document, which just outlines some of the challenges with data sharing, what we envision as the benefits if we share the data, and what needed to be in place. And 2 critical outputs from the discussions and the technical groups, etc., I think were the proposal to establish a Pan-African platform for data sharing, exchange, archiving, and also a centralized network management information system, which I would like to focus on for the rest of my talk. So this is the platform, which we call EGARI, which is an Amharic word, which means to the benefit of sharing, to share and to benefit. That's what the word means. And this was the mission, really, to encourage real-time data exchange, to enable enhanced surveillance for outbreak prevention, detection and response, and to inform disease control and elimination. So as you can see, the continent was divided into the regions. Each region would be like a hub which receives data after sharing within country, and then there will be exchange between the regions and across to a centralized place where everything is managed, curated, QC'd, etc. This is the landing page. If you went on the IGARI platform, before data is shared out of the country, all personal identifiers need to be removed. Data need to be anonymized. Metadata should be summarized to remove any risk of re-identification, etc. The data should be cleaned, and then sharing would only happen with express permission from the countries which own the data. So, as I said, this is the vision, that there would be these regional kind of centers of excellence which pull data from each region, and then the data would then be shared across the continent. And in the end, we envision that there will be dashboards to visualize, to analyze, and then to visualize the data as and when they are shared in. So what IGARI is not, we are not proposing to create another NCBI or any of the GenBank, for example. This is not data backup. We envision that this will happen in the future, but at the moment, there is no capacity to do that. And this is just showing you key timelines of the development. So as I already said, this has been happening since 2021, and there will be a launch in November 2025. Already, the data is, the platform is being prototyped, and data from cholera and Klebsiella has been shared into that. As I already mentioned, we encourage the sharing of the data for surveillance purpose, but we also distinguish between data publication and data sharing. The publication, the two are linked. Publication is essential, but there are risks which need to be mitigated before the data sharing. And here we are focusing on sharing the data for surveillance purpose. The other thing I mentioned, which is the network information management system, will really collect data, store the data, and it captures everything from sample referral to equipment and inventory, etc. And this will be a resource that labs would use at the point of collection, testing, etc. And again, this is just a snapshot showing you the landing page, some of the tabs that are currently in use. And why is this essential? As I've already said, I think a lot of work has been done, but it's all paper, and we need to digitize the process, and we need to take inventory of what is happening. We need to manage supply chain, reagents, etc. And so this will come in to help with all of that, and then enhance QC and checks along the way. So to conclude, future is really bright for our continent. We're doing a lot currently. A lot of labs have been capacitated. There's a lot of sequencing and data generation going on, but there's a lot more to be done. And I think that investment is really key if we're to move platforms to the level that we have seen examples of which have been shared in this meeting. So there are a host of partners, supporters, sponsors, etc. here, but I would like to just focus on the participating members of POSNET Africa, who some of whom are here in the room, and then also members of the Foodborne Technical Working Group, which I was privileged to chair, who also did a lot of work, some of which I've shared here. And thanks for your attention.
Thank you so much for this wonderful talk, Eben. So we come to the last talk of the session, and then we have a panel discussion. So this is Courtney Lane. Courtney is a lead epidemiologist at the Center of Pathogen Genomics and the WHO for AMR, and she has specialized expertise in surveillance of AMR, and we will hear also from your perspective. So,
thanks very much, Adrian. All right, yes. So the topic that I've been asked to speak on today is the importance of integrated public health surveillance in a data-rich environment. will say my background is in field epidemiology. I'm a trained field epidemiologist. So really what I'm going to be talking about is the integration of genomic and really epidemiological data for local public health action. Okay. So I think the need for the integration of genomic and what we tend to term metadata is well recognised. I went back through the talks yesterday just because, you know, it was something that kept coming up, and of the 15 talks, I think probably at least half of them mentioned about how really to make maximum use of genomic data we needed to integrate with metadata. And I think we all understand that, you know, epidemiological, clinical or environmental data are needed to answer some of the questions like, you know, do these cases have a common exposure that could prompt public health action? Or does a new variant cause more severe disease? Or is a new antimicrobial resistance gene making infections harder to treat? But I guess what I'm going to try and argue today is that to maximise actionable data for public health, it isn't just about sort of adding metadata to genomic data. It's really about thinking, in my opinion, about how genomics integrates with an overall public health surveillance system, surveillance and response system. So from objectives to sampling to decision making through to response. So I thought it might be a good idea just to take a little bit of a step back and think about what we mean when we're talking about public health surveillance, or at least what I mean when I'm talking about public health surveillance in this context. And I quite like this framework that Michael Baker from New Zealand has come up with when talking about sort of the different things that we do in public health. When he wrote this, it had nothing to do with pathogen genomics, but really it broke down public health surveillance into sort of reactive public health investigations in response to something, say like an outbreak, where we're really aiming to identify and control risk. And then he broke prospective surveillance down into two different ways. So control-focused, where we're looking to inform control measures, so things like detecting individual cases and outbreaks. And then more strategy-focused surveillance, where we're talking about monitoring trends in disease burden, pathogen types and risk factors. So I've sort of thought about what are some of the uses of pathogen genomics within each of these sort of, within each of these categories, which I've put up there on the slide now. Sorry, I think in copying the slides over, the formatting has gone a little bit weird, but that's okay. So what I thought I would do is just pick out a couple of examples where sort of in our experience, integrating some of this genomic and epidemiological data or integrating genomics into these ongoing surveillance systems has been really useful, both within Victoria and one from overseas as well. So this is the first example. So in July 2024, we had a large outbreak of Legionellosis in Victoria. We ended up with 114 confirmed cases. It was quite severe, around 95% required hospitalisation, and many of those ended up in intensive care. So we did some genomic analysis. We sequenced isolates from some of the cases where an isolate was available. And there was quite a big public health investigation to collect environmental samples as well. And on phylogenetic analysis, it was quite clear that there was one cooling tower that was positive for Legionella, where it was very closely related genomically to the clinical cases. But when we sort of looked at the epidemiology, and I will say, sorry, I didn't do any of this. All of this work is courtesy of Sally Dougal at MDU as well as the Victorian Department of Health. And so this tower was in Western Melbourne, but when we looked at the epidemiology, we could see that the exposures were all sort of east of the tower location, and a lot of these cases were really geographically dispersed, probably more than we would expect from a single tower that was right over in the west of Melbourne. And it was looking at some of those high wind weather patterns that were occurring at the time that sort of crystallised a hypothesis that maybe it was the environmental conditions that were causing some of that easterly movement, more so than we would usually expect. So sort of in this scenario, we've had a reactive public health investigation where we've integrated epidemiological genomic as well as some environmental information about environmental conditions to be able to sort of support or refute an outbreak in this case, and sort of clarify what might have happened where there was some unclear epidemiology. Just a second example is one that some of you might have heard about before, because I've presented on this quite a few times, but it's our prospective surveillance system that we introduced in 2016 for carbapenemase-producing organisms. So for CPOs in Victoria, they are notifiable, and for every case that's identified, we sequence the isolate and we obtain quite detailed travel and hospitalisation history. And the reason for this is really what we call a search and contain strategy around transmission. So we're aiming to identify transmissions when they're really small, to be able to implement control measures and stop further ongoing transmission. And what we've found over time is really that this integrated approach is needed. Oh, okay, a little bit further behind than I thought. Because we've got asymptomatic carriage and long colonisation periods and complex patient movements, which means if we try to use genomic or epidemiological data alone, it's really hard to link cases. Okay, so in this scenario, where we're using the combined genomic and epidemiological data to detect outbreaks early and small for this control-focused surveillance approach. And the last example I just wanted to give is from TB, and Dr. Thinley Dorji, who's here, this is his data, and I think he'll present later on, a little bit more on it later in the week. In which case, you know, in this scenario, they sequenced 163 drug-resistant TB and 53 fully sensitive TB and tuberculosis isolates. And the hypothesis was that they had repeated introductions of drug resistance TB into Bhutan. Sorry, I didn't say this is in Bhutan. With a concentration of transmission in certain occupational groups. But when they did the phylogenetic and cluster analysis and overlaid some of that epidemiological data on occupation, they found that actually there was a large cluster of putative local transmission. So that was sort of an alternate hypothesis to what they had expected. So I guess here, in terms of monitoring trends in pathogens and risk factors, you know, we're really able to integrate genomic, at least Thinley was, integrate genomic and epidemiological data to be able to understand transmission a bit better. Okay, so we've got these really nice little case studies as to why this can be useful, but you know, what are the barriers to doing this more broadly? Because certainly we aren't able to do this for every pathogen, you know, that we have as a notifiable disease or that's under surveillance. And I think a lot of these are very similar to what was discussed yesterday when we were talking about data platforms. They're things like a lack of interoperable data systems, and particularly the complexity of integrating, you know, complex genomic data within traditional epidemiological databases that tend to use categorical data, the lack of harmonised data standards. And what it really means is that a lot of our management but also analysis methods are really manual and resource intensive. We tend to have workforce structures that, you know, create silos, not just of data, but also of people and expertise. You know, there isn't a lot of cross-training across epidemiological and genomic methods. And of course, you know, as we discussed quite a bit yesterday, you know, a lack of perception of benefits or understanding of the value add and the real need for strong governance and trust around a lot of this. Okay, but in my experience, and I don't mean to speak for anyone else here, you know, what are some of the things that we think have worked? Well, I mean, I think starting with lower burden and high consequence pathogens or use cases, you know, in these scenarios, there's often a lot of will to address some of these underlying governance issues and to undertake that really resource intensive data management and analysis processes. Time-limited projects and leveraging research ethics and governance and effort that's put in the research base can be really useful. Building end-to-end integration of expertise. So, you know, really undertaking a process of building capacity of genomic literacy in those public health end users, in those epidemiologists, and really, you know, that early and continual engagement with our public health end users. We've found that co-appointments across public health labs and public health authorities has worked really well as having, you know, greater epidemiological expertise within the public health laboratory. And then I think finally just that, you know, really when thinking about, we tend to have a habit of just sort of going, okay, well, we'll sequence everything and we'll get all the epidemiological data, but really making sure that that integration is appropriate to the objectives, but also the capacity to respond and utilise data. There's no point having, you know, a surveillance system that was very, very sensitive to outbreaks if you're not going to have the capacity to respond to that. So it doesn't have to be all or nothing. Doing subsets or periodic sequencing or periodic integration of data can be more efficient, especially for things that are not as time sensitive, like some of that, you know, looking at ongoing burden and trends. Okay, So final slide. What do I think is the way forward for us? I think a lot of discussion yesterday was around the need for technical solutions, so interoperable bi-directional data flows, and we've sort of done this little bit of a mock-up of if we had, if we were to, when we're trying to, implement this sort of ongoing data sharing and data integration across notifiable diseases and AusTracker, how would that look? What needs to be in one and what needs to be in the other? And sort of see it as a little bit of an overlapping Venn diagram. Not all data always needs to be integrated. We really need translational research to build some of these more kind of robust methodologies to integrate genomic and epidemiological data and to demonstrate that value add. But all of this needs to be underpinned by strong governance mechanisms, purpose-driven data access and sharing, and continued investment in workforce capacity across both labs and public health authorities, and a commitment to collaboration and sustainable resourcing of that. But ultimately, what would be the ideal? It's a seamless flow of information and expertise from the lab bench through to the frontline public health to ensure that our decisions are not just sort of data-informed retrospectively, but really prospectively data-driven. All right.
Thank you very much. You can actually, yes, stay on stage. So we come now to our panel discussion. We are really well in time. And due to this, we will have the panel discussion for the next 40 minutes. So we finish at 10:40. There's more room for tea. And discussion during the break. So everybody is on the right side, okay? Very good. So in this panel discussion, we will address a bit the pro and cons of data sharing on a global level. And I think this is a very important discussion, obviously. I very well remember that during the pandemic, there was once this situation where some new variants were emerging in South Africa, and that really then took a bad turn in the situation that there was a travel ban initiated and so on, which is, viruses are faster than obviously the sequencing was done, and it was already spreading, but there was some severe consequences of this, and I think we should really discuss now a bit the different aspects and viewpoints. And I have a set of prepared questions, but I would really like to engage early on with the audience. So maybe you have questions, and there are microphones around. So please, do you have questions regarding or comments on what you have heard this morning? Yes, please, Ben.
I'm not sure if it's a question or a comment or just a point of sort of tension, but it was great to hear from Taiwan around the use of data for public health, but also the research publications coming out of that data and sort of sharing the data in that research environment. And I guess all the talks had slightly different levels of research sort of leadership. So in Malaysia it's a bit more IMR that's leading that work, and in Australia, it's sort of sitting right in that public health laboratory. And so I guess it's just a comment around the need to ensure that we think about data sharing in a way that facilitates both actions. So if it's driven out of research, it has to, and maybe we could have a comment about how you ensure that you have appropriate public health actions if it's a research organisation driving the genomics, and vice versa if it's a, maybe Courtney could comment on, if it's coming directly out of public health, how do you make sure that data can be utilised for new discoveries as well as just public health actions?
Yeah, any reply to this or thoughts? It should be on, okay. I guess, yeah.
Yeah, really it's a different label about the research and public health. Yeah. Mostly we publish the article more about research, and according to the public health, before we release last news, we have to communicate with the stakeholders of our government, like the animal or agriculture industry and our FDA. Yeah, because we have also to protect our economic industry. Yeah.
Yes, Danielle?
I'll just add to that. So I am a discovery researcher, but I work a lot with the Public Health Reference Laboratory here in Australia and also with collaborators overseas. So I think there's two parts to that. On the research side, you know, when you see publications come through and you're in an editor or reviewer role, there'll be discovery researchers with a really complex, bespoke pipeline that would take weeks to run, and then the bit in the discussion is, and this will be really useful in public health. And I know, you know, Jake, who's the head of Bioinformatics for MDU, does not have weeks to run a complex, bespoke pipeline that may or may not pin down one case. So I think from the research side, it's being having an awareness and engagement with those in public health to what they actually have to do. It took me a long time to get my head around validation and NADA accreditation in Australia. But I think from the public health side, it's also an understanding of what discovery research can do, but also sometimes the time it takes. It can sometimes take a year because you're in the weeds digging around. And then I think having a clear objective of what we're trying to do in the discovery and how it can help public health is achievable, but often the time spans are quite different. If you think like when you have to make a call, Courtney, it's weeks. I'm working on a project with Jane and it has taken slightly longer than that.
And this can also be sometimes frustrating, I believe. So, I mean, we had this huge outbreak of Corynebacterium diphtheria in Europe, and I mean, the news were out, everybody knew about it, like all the public health authorities reacted to it, there were countermeasurements, but it took us like 2 years to actually publish the paper, and yeah, sometimes this is really like you have different timelines in place. I fully agree. Please.
Yes, I was going to say that I fully agree with what you said earlier, that during outbreaks, really, I think the public health focus should be, we want to understand the outbreak, we want to manage it, we want to save lives. And so publication comes second. And particularly in our setting, I think the challenge has been that research institutions are too keen to get a paper out rather than give data that will support the management and save lives. And we've seen a lot of examples of this. So during a big cholera outbreak, which I won't mention, where research institutions already were releasing preprints, while the public health lab were struggling to identify what clone was causing the outbreak. And so I think really the first purpose of sharing data is that we want to help with surveillance, and the mistrust around the use of the data is what has really caused the reticence in our part of the world from people sharing the data, because everywhere you go, people go like, if I share the data, somebody will rush and publish it. And the benefit that I really want is not coming. So I think we should really be balanced here. Publication is great for us as academics, but it's not the primary thing that the public health labs need. And the people just need management, they need help, and so on. And so while we want to publish and let the world know what's going on, I think that we really should be careful not to go in with the aim, I want to scoop data, I want to publish, but I want to help and save lives. We really should balance that. Otherwise, we will still have issues of data not being shared for these kind of things.
Okay, if I can follow up to Prof. Ben. It is true, because in Malaysia, whole genome sequencing is led by IMR, because we are the research institute, and we know our sister public health laboratory, they have MySeq. But currently, this is important. We need to really understand how the healthcare system in a particular country might not be similar to others. The good things about Malaysia, healthcare is governed at federal level. We have Ministry of Health, and within Ministry of Health, we have Research Institute and Public Health. And the good things about Ministry of Health in Malaysia, I think the good points about us is the flexibility. We have public health epidemiologists in IMR. And one of our public health epidemiologists is now the director of National Public Health Laboratories. He's appointed to be. That's how the cooperation, the potential that we can see to bring forward. Currently, the National Public Health Laboratories, their bread and butter is to do diagnostic surveillance. They don't have the enough resources to accepting isolates for whole genome sequencing. And to do whole genome sequencing at more routine scale requires specific money for that. And in Malaysia, with the budget constraint, it is difficult to justify for one. While IMR, we have research, MOH research grant money, we use that particular grant money to justify at least for 3 years, and then from there we start incorporating more isolates, and that's how we start to create more awareness, more interest from the clinician to know, to ensure that they know next generation sequencing is the next PCR, at least for Malaysia.
Okay.
I guess just to add one thing, I think that, in Victoria, we've been in this sort of lucky situation where through a quirk of history, we've ended up with a public health lab that's sort of embedded in a university, which has meant that, there's been a lot of, I guess, close collaboration across researchers as well as those working in public health. But I think even sort of in that setting, we're in, I guess, an environment where there's a really rich public health data that's being collected, both epidemiological as well as all the sequencing that's going on. And one of the, I think, the biggest challenge is even just the real practical sense of, public health people are, sitting there thinking about what do we do? Do we respond? What is the next thing that we need to do? And it's very reactive. And, they're not necessarily going to know, what are the potential things that we could do here to further our methodologies or to really understand what's going on better and to sort of be able to frame some of those research questions. And so, I think a kind of recurring theme for me across all of this is that it's really important to somehow find a way to embed people with some of these, research guilds or genomics or whatever it is within that system a little bit more so that we can sort of cross-pollinate, bring researchers in to help to identify what some of those research questions might be, because we do also have, I guess, a bit of an obligation to make sure that we're using that public health surveillance data for the maximum public good, right? And so, but at the same time, how do researchers know what data there are? Like there's just a bit of a, I guess, a communication gap there that we sort of need to fill.
Okay, excellent. And maybe Peter?
So Adrian, firstly, I really like to comment the other day about looking for the rich terrain between open and closed, and with that in mind, I was challenged recently to think through benefit sharing, because we know that the annex on pathogen access and benefit sharing is currently under negotiation at the World Health Assembly. Okay. And so there are two things that are very clearly on the table. The one is a sharing of medical products. The other is a sharing of money, like, for instance, happens to some extent with PRP framework. Okay, but I think for the speakers, given the range of interest, because the Omicron situation benefited a few researchers while disadvantaging massive numbers of tourist operators, right? So in terms of multilateral especially, I'm thinking, what other benefits are important to share in terms of the if people are sharing data, what do you think they should get back in terms of, yes, obviously academic credit, and obviously we all want money to some extent, but that's more complicated. We can discuss over coffee the mechanisms. Okay, what can you think of? What other mechanisms do you think that need to be shared to increase equity in global pathogen surveillance?
Well, I've got a mic, so I'll make a quick comment and then I'll pass it on. I think first thing that comes to mind is something like vaccines, which you find you need a lot of data to go into identifying targets for vaccines, etc. But then once we get those vaccines, then access in certain parts of the world, particularly our parts of the world, is limited and it's super expensive, etc. So this is, I think, for me, a low-hanging fruit, and first thing that comes to mind in terms of benefits. If we've contributed data and based on the data we've got a vaccine, we should be able to benefit from that, and it should be made accessible, it should be made affordable for those who contributed the data to get the rewards. And I think this is super important in terms of sharing benefits. Then next time, I'm keen to share my data because I know if there was a useful output, we would benefit from that.
It's kind of a quite altruistic way of thinking, I have to say. I mean, in the Nagoya Protocol, for example, when you share a strain, you would want to benefit also economically from that. So Do you think, let's say, if there's an outbreak and you would share the data, should you then also benefit from the incomes of a pharma company, which makes, in the case of SARS, there were billions earned?
Okay, I agree with Dr. Because I think the most peers, like the COVID, if we don't share the data, we will not know how is the strength changed. Then if we share the data, we can do quickly action to protect every region. Yeah, I think it's a benefit.
Okay, I'm coming at it from a pathogen genomic, like that part of the sharing. And I think, so I spoke about typhi, but I think, if people try and build capacity to sequence, a lot of the time you need to have that infrastructure of how to interpret the data. And so I think, the typhi was one example of building momentum, and a publication does arise. But I think there's a couple of things. One, it's bringing together, obviously, the researchers in the different working groups. In the working groups, for example, we have a lot of people that have no genomic data to contribute, but they are actively interested or building capacity in that space. They are very engaged in turning up to the working group meetings where we talk about how we're approaching the analysis, how we're interpreting the analysis. And to them, they're like, this is great. I'm learning how you guys are talking about it, but also what you're doing behind the scenes. So then when they build capacity and have their own genomic data, they already kind of know where the community broadly is at. I think I often look at things by bug, and I think there will be, I could see a space where there's increasingly, for the big priority pathogens, developing that knowledge framework where, for instance, if you have a Klebsiella genome, there's platforms for that, for like, this is how you should be interpreting it. For different pathogens, Celeste highlighted yesterday with rotavirus that the lack of those frameworks mean that people can't actually do anything in that space. So I think the give back there is not only getting a publication, but having that engagement so you build like the skills to do the research and implement, and also disseminate those frameworks so that people aren't constantly having to be like, what do I do if I have a genome X? There's a kind of a consensus that is open access, potentially could be hosted in a way that's almost like a SOP, like a standard operating protocol of, you've got this genome, these are the recommended workflows. And I think that then saves time in a public health capacity space, because people aren't having to think so much, what do I do now? It's, the discovery knowledge-based people have worked in this space and shared that back and made it open access. Yeah.
Almost sounds a bit like a game theory problem here. There was a recent paper in Science about a game theory of data sharing, so I would really recommend to read this, because it's also what do you gain in winning the game, basically, by sharing data. There are a couple of raised hands I see. Maybe you go first, and then we give the microphone to you. So there's already a microphone. You can.
Hi, so I want to hear from each of you, and I'll make this short. And this can be one of those kind of fun questions, but if you could remove one obstacle in your work, what would you choose and why?
Which way?
What would make kind of your work easier and how you're applying genomics for public health? And it could be anything, right? It could be we could change that law, increase data sharing, right? Like, let's try and limit it to, like, of course, more money would be helpful too, but like, what really big challenge that you really wish wasn't there?
Can I go for a petty challenge?
Yes.
That's just incredibly frustrating.
Yes.
Dates. In the metadata, you're trying to do a temporal snapshot, and the number of times that you constantly, and you never save the code, but constantly having to get that sorted. I'm sorry, the US, your date system messes with me every time. But I think that's just one of those things where you're trying to process it quickly. It's really petty, I know, but it drives me mental every time.
It's perfect. That's kind of exactly, I think, what I was looking to hear. So I'm interested in the other comments as well.
I'm going to give two answers. So my first one is from...
Oh, sorry, I was talking.
Do I really need it? Okay, I'll use it. The first one is from Victoria, and I think that it is... the technical interoperability of data. So, or the lack of there, I guess, is what I would remove. So I think in Victoria, we've managed, particularly with our public health colleagues, to develop a lot of trust, a lot of the governance that's required, but the amount of time that is spent data cleaning, linkage of data, all of that is just astronomical. And if we could remove that barrier, I think we would be able to progress what we are doing much faster. But from a secondary point of view, with my other hat on, which is a lot of the international work I do, I would say it is the, I guess, maybe lack of interest or investment from public health decision makers, where, we have some really keen public health labs who are really developing capacity, but being able to get engagement from public health decision makers to understand what their objectives are is really challenging. So maybe it's two answers, sorry.
I think for me, it will be the bureaucracy in the system. There's just too many levels of approval, and it makes the work super slow. Like, you need to talk to this person, and it takes ages to get anything done. And there's an outbreak, but you can't take action because there's just too many things in the cog that make it frustrating to get anything done. So that, for me, will be the first thing to go.
And of course, then afterwards, right? Like, they'd be like, well, why did it take you so long to solve it? And you're like,
yeah, the bureaucracy is really an issue. I agree with you. I mean, I just had a recent episode. We had the ethical approval, we had the contract, and then somebody in the consortium said, should we not all agree again on it? And then people started to email, yes, I approve this. And then, no, we have actually the basis and everything. Why do you have to reapprove now something we agreed upon? And this is really a bit weird that we kind of try to slow down ourselves, even though everything is set. Okay, really good. Now, please, yes.
So, data sharing and governance, these are a very long-standing, problem. Data governance, governance is important, but when it comes to data, public health data, basically, we have to rethink if we basically follow the kind of stringent kind of rule to share, because this public health data are like not like anything else which you can make the use of for economic interest. This is public health data. Like I like the policy of Taiwan colleague, open access. But what makes it difficult is like some of the kind of data scavengers, they might make the use of this data for their own interest. And this is why each of the countries that basically produce the data, they believe that these are their valuable treasure. They can, but unfortunately, this data actually are of no use to them. Basically, each of us, we can think about this like back at our own place. Like, we will leave behind the data, leaving behind the data basically. And when this data are basically not helping anyone, but just becoming a treasure. Which is, I don't know what is the, benefit of it. So each of us, we should rethink that whether we should stick to this kind of governance policy. I don't like it. Back at home, I also have this same kind of governance policy. But as an individual, I hate this. I hate this because the public health data has immense public health importance. It can save life. I'm just abstracting, public health, and just not allowing the data to be translated to produce some vaccine or any kind of intervention material. So this is so much important. We should think, and we should do something from our own standing. I know we have the upper level ministry and government, but when we sit with them, I sit with my government and I speak loudly that we should not abstract. Like, I was sharing data with the Welcome colleagues. We had a project and we generated a lot of data, and we actually were kind of requiring the Nagoya Protocol compliance. So this was a long kind of bitter experience. So I spoke up and I really acted, and it helped. So this is what I wanted to share with you, that each of us should play our role from our own standing even if we are in the mid-level or in the lower level, just this is what is needed. Thank you.
Quick reaction?
Yes, please.
Yeah, I
please.
just wanted to react quickly. So 100% I feel your sentiments and I'm with you, but I would like to play the devil's advocate. So I think we shouldn't underestimate the importance of data ownership, you know, like the pattern from, I speak from Africa CDC and African continent, the member countries own the data. Africa CDC is just a curator of the data that is deposited into, let's say, a centralized platform. But you need express permission from each of the member states before you can do anything with the data, including sharing to third party. What I think is beautiful and is being done by Africa PGI is in peacetime, if you like, already have an agreement in place with the member countries of the African Union at the governmental level to share data, so that when there's an outbreak and there's a need for data to be exchanged, we don't then begin to go through the process of, you know, seeking ethics and whatnot, because we've already agreed that we need this data to be shared in real time during outbreaks. But I think we shouldn't downplay the fact that data is super important. Even if I don't use it for anything, I don't want you to use it without my permission. And we would all say the same thing about anything we own, including my date of birth or my phone number or whatever it is. So I think having that kind of discussion in advance of outbreaks is what will get us there. Then when we need the data, we already have arrangements in place, agreements in place, and we will abide by that, and there's that trust, and so the data will flow. That would just be my caution here.
So having not shared, they are deceiving themselves.
Yeah, it's an interesting point. I mean, the right of the individual versus you know, the greater good for the population, and that generates a lot of tensions. And we as a society have to discuss that, you know, what is important for us. I agree. Good. More reactions on this? Further questions? Oh, yes, please, please.
I was thinking that last time we have problem with reviewer, general reviewer, right? Now we have credits for reviewer, of which we have a list of reviewer. For probably we have similar system for data that is being utilized for an outbreak or journal articles, so that we know the institute being acknowledged properly with a matrix that is tangible. I'm not sure. Another perspective, we don't have this problem as yet, but start thinking that the hospital that contribute the isolates to the lab might be asking what they can be benefited from, because sending an isolates, putting some metadata in the worksheet, so requires some effort for them. I'm not sure. Perhaps I would like also to know how the other labs are acknowledging this. But for MR, we plan to let the sequence depositor to name the strains, but we are not sure how complicated things will be.
Yes,
Hi.
please, Jake.
Thanks for all the talks, everyone. I love data sharing. As much as I know Ben, our director, I annoy him about sharing more and more data all the time. Yeah, I know. It's been great. And I think, Danielle, you flagged a really important aspect of momentum when it comes to sharing data. I think once people get used to sharing data, they're more likely to share more. And Australia's history of sharing genomic data has actually been relatively poor, despite us having really established sequence surveillance programs. So before COVID, as an entire country encompassing all the research that's ever been submitted to NCBI, we only shared about 30,000 genomes entirely in the history of genome sequencing. But through a bunch of our national programs that we've established and shared, we've shared more than that data in the last 12 months. And I think a lot of the data sharing comes from, or the lack of, comes from fear and unknown. But once we started showing people, and we really took the lead in Victoria, we just started uploading a lot of stuff. And then all the other states and all the other public health labs realised there was actually nothing wrong with that. And then once South Australia came on board, they started sharing really fast, and then that momentum started spreading. And now we're able to do almost national surveillance through NCBI pathogen detection portal. And so I guess, are there any strategies from anyone on the panel, that maybe you can utilize that momentum and maybe take that first step from your jurisdiction to try and convince others to do the same.
I can start. I would say there's two parts. One, with the consortium that I spoke about, that memorandum of understanding is quite significant, I think, to people sharing the data ahead of time. It also means that, you know, no one's breached it, obviously, but I think there would be a lot of repercussions because the whole community's together. So I think it creates that momentum there of best practice. I know from a research perspective, I am conscious of some of the issues that were raised by Eben. So, for instance, in my mind, I'm like, high-income country, Australia, USA, Europe, you're free for all. Your data's there, we'll go for it. But if it's data from a low to middle income country submitting and I can't find an associated publication, I normally deliberately exclude it just because I think someone could be working on it, and the effort that often goes into getting some of those genomes, especially in the early stages of capacity building. So that's my personal approach from the research side. I feel like the high-income countries submit so much and throw it out that I'm not scooping anyone there, but I really am conscious of some of the issues that you raised, Eben.
Yeah, I think a lot of communities of practice are emerging in this space, and that has been very helpful to bring transparency. Like, you meet with people from all over the world, we talk about what we're using the data for and what are the benefits, and when we sit in some of those discussions, we get reassured. From an African perspective, I think there's a lack of trust. If I have to deposit my data in a repository that is outside the continent, it doesn't matter whatever you said you won't do with the data is going outside of my country, it's going outside of my continent, it's not owned by an African, and so culturally there is a lot of reticence there. And you see this when you talk to people. You show them examples of benefits, you show them in blueprints, you know, statements about privacy of the data, etc., but they just don't want to do that because it's not on the continent. And so I think one of the ways that we can remove some of the barriers is that we need investments, we need co-leadership, we need Africa-aware, kind of Africa-led projects and platforms that will give confidence. So we need to have investment and do some of these things ourselves. It's not enough you've built a platform in Norway or in Australia and you want me to use it because it's safe and whatnot. I need to be able to have a link on my continent where I have my own technician or bioinformatician involved, then I can trust that, yes, you're going to look out for my interests. So I think that's something to think about as well.
It's a very, very interesting point. So for genome sequencing data in Switzerland, I can quickly comment, that's not so much an issue. So the sequencing data, we are fine by having this, you know, anywhere. But with patient healthcare data, so we had a huge incentive in Switzerland over the past 8 years, and we collected from close to 100,000 intensive care unit patients time series data, so from monitors, you know, all the lab values, etc. And there was really a debate, where should we actually store this? And we decided not even to go outside of Switzerland with this. So we really wanted to have full control of the data. And we have to face a bit the reality. I mean, 40% of the cloud-based data is owned by Amazon. I mean, this is also crazy that we have these hyper data centers, 540 hyper data scale centers, and they are like, half of it is in the US, a third of it is in China, and there's basically no large centers in low and middle income countries. That's also a critical aspect when you think about the petabyte of data you're going to produce. Where is this stored down the road? Okay, I think there are further comments from Tim. Tim, yes, please.
Thanks everyone for the panel for the presentations and the discussion, and I think they're really interesting points made across the piece. I think the best examples of data sharing often come within networks where there's a clear use case and trust between the members of the networks to share data. And I think we've seen that, traditionally in high-income research settings, but I think it's, it's really encouraging to see that now with local capacity, like the cholera examples in Africa, where there's clear incentives to public health questions to share data. And I think when we look at, from a global perspective, we need to really be able to articulate, what is the public health benefit from sharing the data, and if there is no and if it's an altruistic part, that there is no local public health benefit, I'm not going to understand my outbreak any better, or I don't have the capacity or the intervention to do the thing that you can do in high income. That should also be articulated. And I think it's often we speak very blanketly about data sharing, where there's nuances to every pathogen and every public health question, and I'd like to see those use cases articulated further. And just the second point I think is key is the data can only be shared if there's capacity to produce that data and sustainable capacity to produce that data. So I think when we think about benefit sharing, there's obviously concepts around sharing countermeasures or monetary contributions, but I think we should think, how could that feed back into sustainable capacity development, where the agency is on the producer of the data and on the local priorities of that data, as it also benefits the global surveillance. I think the final point, and I'm kind of deep in it, is the pandemic treaty and the access and benefit system. So this is, I think, a positive step towards taking a multilateral thinking around sharing data, but it is very much only applicable to pathogens of pandemic potential, which has not been super clearly defined, but there's plenty of things which we interested in sharing data that aren't going to fall into that. So I think as a community, we're still going to have to work through how the current laws and treaties like Nagoya, which don't specifically, that they're for bilateral agreements, they don't specifically think so much around sequencing data. So I think there's still a lot of work to do in the community, and I think I would ask for the people who are with these pathogen specific networks, really publish the papers telling the stories around the use cases as much as the kind of the public health outputs, because I think it's really important. But I will stop there.
Ben?
Unless there's a response to that, I just wanted to make one quick point on technological solutions. So like in Australia, we've talked a little bit about the platform being used here, but a key part of that is the permission system, and what the data ownership stays with the laboratory that uploads the data. There's a lot of permission systems to allow sharing and those sort of things. So it gets back a bit to what Eben's saying, that I think technological platform solutions can be good, and I'm looking forward to Tim's document on the characteristics of data platforms and how they should look going forward. But that's one of the key things, I think. And just also to comment that we've got that same issue that people don't want to hand over data; they want to be part of the next step. And so, in Australia, at least, every state and territory, we are 8 mini countries really. Every state and territory has a role or can have a role in the next steps and the analysis, and everyone only trusts their own bioinformatician. So, I think those things can be helpful as well as part of the process of moving forward.
Mm-hmm. There really seems to be a more progressed understanding about the value of data, and data is a resource, and that you want to be involved continuously. If you share data, you just not give it away and then somebody else takes it over.
I was just going to just agree with what Tim was saying about really needing to articulate what the use case is and what the benefit is. I think we've talked a lot about public data sharing, and I find that often when we talk about data sharing, that's where the conversation immediately goes. But even if we're talking about within closed networks between different sectors, or in sort of a federated system like Austraca, things like that, people are always reluctant to share data unless they can see the benefit and the use case of it. So it's just, I guess, underscoring that necessity to articulate those, but also to lead with examples that you can then build on to sort of demonstrate what the evidence is for I guess, benefit.
Okay, we are very close to the end. I want us now to imagine maybe, okay, last question over there, then I wrap up. Maybe we can give a microphone. Just a second.
It's not really a question, it's more a contribution, I guess. I'm not in public health. I'm a microbial ecologist, and within the field of microbial ecology, we do share a lot of data very quickly before publication, a lot of the time. And so we've had a lot of conversations around appropriate and poor use of other people's data for quite a while now. And quite a few of us have gotten together and written a paper that will come out imminently on the idea around a data, what are we calling it? The data reuse information tag, which will be associated with an ORCID identifier. And it's an idea that may not apply 100% to this group, but it's one way that researchers can talk to each other about how we want our data to be used amongst ourselves and what it can and cannot be used for, and whether or not we want to be contacted or not for how that data will then be used. And so it, in the next month that paper will come out, and it's something that this group may also benefit from too.
Thank you very much for that comment. Now, let's imagine we fast forward a few months. It's Christmas. You're opening your gift, and in the gift box of data sharing, what is in there? Maybe each of the panelists can say what is their data gift. Or a wish maybe you have. I don't know.
I'll kick it off. All right. I'm going to say an alignment of expectations. I feel like a lot of the time, when there's people have used similar language but arrived at a different viewpoint of what they should be getting out causes a lot of trouble. So I think having everyone on the same page for the whole time would be beautiful. That's mine.
Okay, one happy family. Very good.
Yeah, I hope the gift I receive is respect and trust. If we trust everyone, then we can easily to share our data, and don't worry to use it.
A very important message in the increasingly separating world. I fully agree with you. Trusting each other is absolutely key, yes?
I was going to talk about trust, but I feel like that's been adequately covered, so I'm going to go with something else, because we can share our Christmas presents, I assume. It would probably be look really interoperable data platforms that are, locally deployable, so data doesn't necessarily need to head offshore or whatever, that have secure and tiered access requirements so that, people can have purpose-driven data access and we can maximize the benefit within a trusted network. I know a lot of those are sort of already under development, but I think, there's quite a bit of work, so, skipping to the end and they're done and they're ready for me and all wrapped in a bow.
Very nice.
I think I would like to wake up to our governments putting their money where their mouth is, and a bit of equity in the system, but more so investment from our own governments to help us solve the many questions that we have on our continent.
Okay, it's awesome. We have harmony, we have money, we have comparable things. Maybe one last word from...
I already got the best present today, experiences from others about how data sharing looks like, and we can learn from your experience how Malaysia would tailor our own system. But for Christmas, we really hope that we can get Illumina I100 so that we can create more data to be shared. Thank you.
Awesome. Okay, so maybe you get a Christmas gift from them. Who knows? Okay, thanks a lot for the awesome discussion and also the interaction. And with this, I would like to close the session. And now it's tea time. Yes.
Thanks, everyone. We'll be back here at 11:10, please.
Thanks, everyone. We're going to start the next session now. So if you could just sit down and hopefully you can find a seat. We seem to be quite a few more people here today, which is good. So we've just got another single talk now before we have the sort of interactive workshop. And our speaker is Michael Abdo from Illumina, and he's going to talk about sort of the future, sort of glimpse into the future of accessible genomic technologies, which we're all very interested in. Thanks, Michael.
The Christmas gifts, yes.
The Christmas gift. Can you figure it out? Because I can't.
So no pressure then. Not only am I the only talk after morning tea, but apparently I'm giving gifts left, right, and centre. Look, in all honesty, don't forget for your afternoon tea, there are Tim Tams still on the table there. I haven't seen any Tim Tam straws yet, and we have the Lego sets as well. So if you have kids at home, please do come across and grab a set. Now, another caveat, I guess, to the start of my presentation is I actually do want to take a moment to leverage on a theme that Mark suggested yesterday and dream a bit. Because I fear that if we don't consider all possibilities, we actually aren't going to leverage the potential of genomic technology as it stands today and what it can mean tomorrow. Now, having said that, my crystal ball is no more or less opaque than anyone else in this room. And so I really just put this out for thought, for conversation, perhaps to simulate ideas for the workshop immediately after my talk. And to give that some context, in my hand, I can control my lawnmower back in Western Australia. I can go to a restaurant in the outskirts of Tokyo and get recommendations as to what other patrons are ordering, a restaurant I've never been to before in my life. I can go to Taiwan and get emergency warnings around earthquakes or other areas in Southeast Asia around tsunamis. So if I have in my hand the potential for such exploration, such leverage of data, what is the potential of public health and what is the scope of genomics with that backdrop? Now, we've already seen in regards to the methodologies for pathogen surveillance how NGS has impacted this. Originally, we would have approached an outbreak or something to that effect, focusing on a single pathogen, possibly even a single gene within a pathogen using PCR or Sanger sequencing. And yet today, we can routinely sequence whole genomes. And the generation of those whole genomes has now enabled us to move on in terms of our pathogen surveillance. And we can look at syndromes and multiple pathogens simultaneously, and pathogens that are resident in all weird and wonderful environmental samples and the likes, really broadening the scope of our surveillance and the resolution of our surveillance strategies. And increasingly, we can take that even further to now explore hypothesis-free investigation for really acute cases and emerging threats, simply because of the massively parallel nature of NGS as a technology. Having said that, we've heard over the course of yesterday and even this morning that a number of challenges exist. Now, I can't use a pointer on the screen, but if we think to the left, what we haven't heard a lot about are the biological challenges and limitations. And indeed, whether it's contamination, whether it's the biomass, whether it's even the diversity of the sampling types, consideration of these and exploration as to how we address these in the future really will inform the surveillance strategies, particularly when you are looking at the robustness in regional and remote communities, when you are looking at surveillance strategies for communities that perhaps aren't exposed to the sanitation that the global north takes for granted. If we look to the far right, we've perhaps already forgotten how simple things like supply chain logistics became a profound limitation for genomic surveillance during the pandemic, and how those challenges exist and continue to exist in low to middle income countries. Now, yesterday during lunch, Gavin had an opportunity to talk about the MiSeq i100 and the innovations in our technology embedded into that instrument, and how that allows you to sequence faster, easier, simpler, and the likes. Now, to a degree, all those elements, in both a positive and a negative fashion, impact on one simple fact around the data generated, and that's the utility of that data generated. And I'd actually like to challenge you in this room to not view the utility from your perspective, but from the perspective of the ultimate beneficiaries of public health. And that could be a community like here in Melbourne in the midst of lockdown, or communities in informal settlements across the continent of Africa. For them, your considerations, as valid as they are around the ethics and the clinical validity, analytical sensitivity and the likes, are important, but do they outweigh the immediate benefits that could come from this information and your activities? And so perhaps there's not a perfect system, and perhaps we don't have to dot all the I's and cross all the T's as we move into the future. And these things can happen in parallel, and we can be prepared to make mistakes, albeit controlled, for ultimately leveraging the benefit. Now, that picture is actually sort of exemplified or illustrated in this concept. And if we consider public health outcome by a single measure, that of mortality, and we look at the epidemiological transition of the global north over the last few hundred years, we can see how there's been a transition between public health concerns from communicable disease and those of non-communicable disease. I think there was a definition earlier this morning where we focused on public health as communicable disease. Well, from our perspective, from a genomics perspective, it's actually both. Your sequencer does not care if it's genetic disease or a pathogen that you are sequencing. And perhaps in communities subjected to, say, malaria, sequencing for thalassemia is just as critical to the outcomes as sequencing for the pathogen itself. Now, over the last few hundred years, we know that in nomadic times, the bulk of the mortality was contributed by either communicable disease or physical insult. And unfortunately, for time immemorial, people will still trip over, still get run over by a bus, sadly. And so that will never be removed. But we have seen changes to the balance between the communicable and non-communicable disease. The agricultural revolution brought with it much greater access to improved nutrition. Didn't fundamentally change that balance, though, in terms of public health, because we couldn't distribute that. The Industrial Revolution actually saw worsening, and this is our best measure as to the profound impact of urbanization and rural-urban drift, and the importance in maintaining safe rural communities as well as safe cities. And then, of course, we've moved into the modern era, and we've seen many countries already transition past the burden of communicable disease to non-communicable disease, but we've also recognized that this is not a linear transition. There are blips along the way, whether it be through pandemics, most recently, or the last century, whether it be through conflict, and whether it be through advances in other areas of medical science around vaccination and the likes, or even school nutrition programs. Now, of course, that brings us to the genomics era. And here, if we continue to consider the global north, we can see the profound impact in areas like oncology that precision medicine has made in improved outcomes for oncology patients. We can also see, and we've witnessed in the presentations over the last day and a half, the impact with regards to antimicrobial resistance and stewardship of antibiotics that genomics can have. When we consider the future, though, we're actually now on the cusp of the multi-omics era. There was a paper published actually just in the last month out of the Netherlands in Nature Medicine, where using Illumina technology, they were not only looking at the pathogen itself, but they were considering the morbidities associated with drug resistance in HIV. And to do this, they did genome-wide association studies in the population. They did methylation analysis, transcriptomics of both the pathogen and the host. And then they also looked at protein expression. And when we think to the future of genomic technologies, you may or may not realize that you can sequence proteins on your Illumina platform already, today. And so I challenge you to think beyond the constraints of merely sequencing a genome. Now your data should be multi-layered, multifactorial, and because that gives you the opportunity to potentially look at asymptomatic cases, latent disease, and really drive a profound and accelerated change in public health outcomes. So if we then consider that same epidemiological transition for the global south, communities can place themselves either to the left, still burdened by a significant proportion of mortality through communicable disease, some going through a transition as the middle class within their economies burdens and is subjected to non-communicable disease and the mortality associated with that, and hopefully moving towards the far right. And I put it to you that the future of genomics is accelerating the rate of change through that epidemiological transition. And it's our responsibility, really, as to how we leverage that data to achieve that. And so, somewhat predictably, but at least consistent with the conversation of the last day and a half, it is that data sharing is key to this. But what I would challenge you is that missing in this room are a number of those key stakeholders on the left. Because when we consider the utility of the data, it actually is vastly different for those stakeholders. I think during yesterday's panel, there was a point made around perhaps we should define data before we actually start the conversation. And if you walk in the shoes of those other stakeholder groups, their definition with regards to data utility is vastly different to ours. But they also have a key role to play in the sustainability of programs outside of the crucible of a pandemic. And so funding may come from pharmaceutical industries who are currently providing vaccines or antivirals for some of the endemic diseases that you are focused on. And that alone would be a profound change in the way we address the public health outcomes. Of course, ministries of health can assist, should drive some of the legislature, some of the standardization that has been a topic of conversation during the last day and a half. All of that, though, ultimately leads to informed decisions being made. And again, I stress from our perspective, the future is not to segregate between concerns of communicable disease and non-communicable disease, but look to global health outcomes whereby genomics is agnostic as a technology. And again, if one's looking to sustainability, implementing those strategies across the paradigm potentially makes the value proposition, the economics of it, far easier to address, far easier to influence when considering funding bodies, governments, and the likes. The ultimate goal being profound change to global health benefits. And so at this point, I may have disappointed some because I haven't revealed Christmas presents or Easter eggs as opposed to what's coming up in the Illumina R&D pipeline. I'm afraid. I'm afraid there are people far higher up in the organization that held that information, and it's not me. Suffice to say, I'm quite confident that the rate of change and innovation is going to continue within Illumina and with the industry at large, and dare I say it, driven a lot by you in this room. What I would argue, though, the future is moving from a reactive response in regards to leveraging or the utility of this data to something that is proactive, something that is holistic in regards to surveillance. Consider the vectors at the same time as you consider the pathogen. Consider the host at the same time as you consider the pathogen. And consider communicable and non-communicable diseases at the same time. Hopefully that gives you some inspiration. Perhaps off of the recording, we can talk about other technologies or more specific things that might be coming out. Suffice to say, do keep your ear to the World Wide Web because there are some really exciting announcements coming from Illumina. And I am not kidding, you can sequence protein today on your Illumina sequencer, which gives you a profound additional data set. But for now, thank you for the opportunity and do enjoy the animals this afternoon.
Thanks very much, Michael. Yeah, I really appreciate the way that you actually sort of tailored your talk to what's been going on here. That's nice to see that, and also to the long-term relationship that we've had with Illumina ever since I was part of purchasing the first GA1 in Australia back at 2008, I think it was. So, yeah, really appreciate it, and yeah, food for thought. Thanks. What was I going to say? I forgot. Yeah, I'm going to hand over to Tim Dorman. Just like I did yesterday, Tim's coming back. Tim's going to run a workshop. Sorry? The return of term. And I think what I was thinking about was Michael's offhand comment about sequencing protein.
All right, thanks everybody. So this is the last thing you'll do today in terms of your scientific input. So hopefully we can bring a lot of energy into some of these discussions. So I really appreciate the organisers' opportunity to use these distinguished guests of GMI to help WHO think through how we move forward in the pathogen genomics space. So I'm just going to do a few slides to to set the scene, and then I'll explain kind of the activity that we're going to be doing for the next hour or so. So, as I mentioned yesterday, in 2022, WHO released a genomic surveillance strategy with 5 objectives to strengthen national and global surveillance with a metric that is equitable access to genomic surveillance for all countries. And so now we're in 2025, we're 3 years into this strategy, and these were the 5-level objectives that were going to help us get there. So improve access to tools for better geographic representation, strengthen the workforce delivery, speed and scale, maximize connectivity in the broader surveillance architecture, maintain a readiness posture for emergency, and enhance data sharing for local to global health, public health decision making. So what we wanted to do within the IPSN network is take a moment to reflect how are we doing against the objectives 3 years in. What have we learned in the last 3 years? Do we need to pivot our attention or prioritize certain things to ensure that in 2030 the global genomic strategy meets this objective? So what do we need to keep this on track? Oh no, that's my computer. What do we need to keep this on track? So we took the opportunity to convene a workshop in June this year. You can see some of the participants of the workshop there, with the aim to develop a roadmap to identify some short and medium and long-term milestones to support this strategy. And the workshop was 2 days. We started with identifying the gaps that the people identified with in terms of the end-to-end process of pathogenomic surveillance, from sample to data, data to insight, insight to policy. And then we asked them to identify, well, what enablers will help mitigate or fill those gaps, and then identify some deliverables that we could work towards for each of those enablers. And basically, there's kind of 3 high-level priority outcomes, which I mentioned yesterday. So this is the institutionalization of pathogenomics through tools, policies, networks, and frameworks. So how do we really embed pathogenomics as a key public health capacity and component? Priority outcome around investment alignment, so around funding landscape, but also around driving the evidence for evidence-based advocacy. So thinking about how, when we talk to our funders or our ministries of health around investments in genomics, what's the evidence base we can provide for that? And finally, a piece around coordinated data systems. So the workshop, as I said, we looked across sample to data, data to insight, insight to policy, so thinking of these different components of the pathogenomic surveillance. And we considered kind of 4 thematic areas of interest. So equity, access and capacity, governance, ethics and trust, sustainability and resilience, and interoperability and standards. And so for each of those, the gaps were identified, the enablers that's going to help us overcome those gaps, and then some deliverables. So you'll see dotted around the room something that looks kind of similar to this. So for each of the thematic areas, so here we have governance, ethics and trust, 4 prioritized objectives were identified, and then a set of deliverables associated with them. And I'm not going to go into them all in any detail now, but you can see one of the objectives was to support the adoption of global genomics guidance into national planning. And then the deliverable was a comprehensive implementation toolkit with guidance, tools, and best practices. And another example for it could be ensure lawful, ethical, and secure data generation and sharing. And then the deliverable here would be national policies, like we've heard many of these national policies being developed, regulations with governance roles and compliance mechanisms. So that's the deliverable here. And then so for each of these thematic areas, we have 4 objectives and deliverables associated with them. So this was the outcome of the workshop. We have these 16 deliverables, and now we want to take the opportunity with you guys to socialize this information, and for you guys to help us think through what are some of the risks and some of the missing pieces that we might still be looking for. All right, so what specifically are we going to do today? So today we're going to contribute to the roadmap. We're going to perform what we call a pre-mortem on the identified deliverables. And so a pre-mortem is basically before the activities start, we think together about, what are the risks that are going to cause this deliverable not to be met? What are the risks that are going to mean this is going to fail? So for each of the deliverables, you're going to take some time to set aside what are the risks that are going to stop this deliverable happening. But we also want you to think about what are the opportunities also in this area. And maybe you can take some inspiration from the previous talk about thinking, if I'm thinking what other things holistically in the life science space might be we can take from as learning for this particular deliverable, for example, or what other activities could be aligned to this particular deliverable. And then we want you to think about how we're going to overcome some of these risks. So specifically, the objectives, just to be clear, so first we're going to identify the risks and opportunities for each deliverable, and then propose solutions or mitigations for the global community to consider. And then you're going to help us identify who can help with these mitigations, who can help overcome these challenges. So this is going to be an incredibly useful, hopefully useful exercise for the development of the roadmap. We're in our early stages. We had our initial workshop. This is one step on the journey of hopefully having something to publish next year to the global community to help us all have an articulated community set of activities that I think we can articulate to funders and ministries of health. Okay, so we're now ready to begin. So let me just take 1 second to pull up. Okay, so you can see in the corners of the room, so we have these sheets which summarize for each of the groups the objectives and deliverables. So what I'd like to ask you to do in 1 second is find a theme that you're perhaps interested in, and split, hopefully relatively equally, if you could, into each of the themes. So congregate around each of the groups. So we've got 8 groups. So each of the themes are duplicated. I think we have 4 in this room and then 4 towards the outside. And if you have a pen or something to write with, to take with you, we have some pens, but I've heard the message is maybe not enough pens for everyone. So if you have any writing equipment, it would be useful to bring that with you. So what you're going to do is you're going to think through the risk. We're going to use Post-it notes, and you have a facilitator to identify them, stick them on the board, to discuss them. And then we'll spend some time going through those risks you've identified and identify some mitigation strategies and who's going to help us do that. So greatly appreciate your efforts and enthusiasm with this task. You have a facilitator on each of the stations. So yeah, thank you very much. If there's any perhaps clarifying questions to start with, or otherwise we can begin the discussions. No.
All right. So if everyone could find which one you want to be in this room first, and then we'll half the group from there and we'll take half outside. That'll make it easier. So if you want to look at which theme you're interested in and stand at that theme, then the second facilitator will come and split the group. So on the left-hand side of the room, we have governance, ethics, and trust, or equity, access, and capacity. On this side, we have sustainability and system resilience, interoperability, and standards. Good hustle, everyone. Move the feet. All right, so you're going to split up?
All right,
While
thanks.
Samia in here, presenting.
So I think some more people over on governance would be appreciated.
It's just there, isn't it?
Oh, yeah, I want to say, great.
Hey, Tim.
Yeah, Tim.
I don't want to have to say this, but we're not here and we're not participating. So don't
So you can walk around it.
No, thanks, Mark.
Too many big words.
Tells you what you need to do.
To be competent.
Yeah.
Angela, can you join your group over here?
So we're just organising the room. Thanks for your patience. So there's another interoperability over there.
Oh, there is redundancy.
Yeah.
No, I don't forget the service. So we're here. Yeah, so there's a split group and then we'll come back together at the end and then as part of it. So the first part of it, we're just going to look at these objectives and then we're going to do a tidbit and you're going to come up with
Yeah. So we're out. All right, guys. I think we're going to start.
What do you want people to do? Are they writing stuff down, are they?
Yeah, so we have these objectives and deliverables. And so the idea is we want to identify risks associated with deliverables. We want to go wrong.
All right, guys, hopefully we've all started. So we're starting now with our facilitators. We're looking at the deliverables, what could go wrong, so identifying the risks and thinking about the opportunities. We have 5 minutes for this one.
Individual notes, they just can do that on the conference booklet or something like that.
It's like so, because like it literally like the room shows this, like
That would be really useful. Yeah, okay. Yeah, so we've been working on one of these things. So the question is like, so what is the reverse proposed by like because everyone's saying not everyone is obvious. Yes. But like I would challenge you to think of like further risks. Yeah. Maybe like you're still on the side. Yeah, exactly. Exactly.
I do have the screwdriver. I just didn't. I packed one, but it didn't have a nut in it. Usually it has a nut. It must be used to produce. No, I have that. I have the screwdriver. I don't have the little one. Oh, the clinic would have it.
Yes. Yeah, it was
So if you haven't already started, if you can begin using the posted notes to recording some of the risks, that would be great.
It's all the yellow ones.
Yeah.
So what is the either risk or opportunity related to this thing? And I mean, I think this is the deliverables for it. So basically, the objectives that and what are the risks? Is it going to happen? No. The risk, yeah, the risk could be related to deliverable where something just we just can't deliver this way. It is not possible to do that.
I'm not sure because we're early.
Switzerland. The problem with the Swiss federal office of public health is that they are administrators, and now they came up with the idea, we need a national surveillance program of plasmids. Now everything is plasmids, and you think like, I'm not sure that this is the biggest problem we have, and it's really, but that's great that we do this kind of, I like this. And what can you do in terms of preventive strategies?
So do you find that could be a risk when such a pandemic that you will have that lockdown? So priorities on the coast and maybe have less I think that
local act global. It's really is.
So maybe that could be a good risk to
I think we still have five more minutes or so. I think now maybe you kind of so maybe is there any themes that So I can see we've captured lots of risks, lots of yellow post-it notes on the whiteboard. So that's great. So I think maybe we can spend five or so more minutes thinking, what are the themes that have come out across the risks? Are there, some risks that, really cross-cut deliverables? So try and think kind of thematically with your facilitator, some of the patterns that have kind of come out from your discussions.
So we do have certified patterns. Some people are but I'm thinking
All right, everybody. So now we've completed identifying the risks against these deliverables. Now we'd like you to solve the problems and identify the mitigations that are going to help with those risks. So facilitators, if you can take what the kind of the key risks that your group have identified, move them to the clean white sheets on the right-hand side, and then ask your group to think about, on the pink post-it notes, what would be the mitigation strategies for those risks? What can be done and who can help?
This is sort of that, the idea is that you build a platform on Azure or Amazon or some platform like that, right? And then you're trapped in that sort of ecosystem. So you have to keep using that tools and so on. If their funding changes, you have to actively see funding moving around. So you have to be different things, right? One is a risk and one is a network or a snake as it gets something that's not operating. So I think about it, it's not connect and then you're sort of trapped. There's a risk.
So that no matter whatever it is, we know that this is a fixed term where
I guess it would be contracts that allow flexibility or something like that. That's trying to prevent that through legislation. Yeah. Yeah.
somewhere else.
Yeah.
Yes, so that's not going to be new for us.
Maybe IPS stuff as well.
Then we can resist some
Yeah.
changes or whatever. That doesn't fit us.
I mean, that's fine.
Like for example, GitHub yesterday was released something about experience. If everybody walks away, I guess.
That's all right. It's almost a unionization or something like that, right?
It's great, but it has
Here we go. You should never be out. Maybe it's the
for instance, just at the moment, Nextdoor you can run on Azure, Nextdoor you can run on Amazon. Because that layer, technical layer allows possibilities. Therefore, if we sustain those layers, yeah, I think that's
because remember we've been talking about standardization and all of that. So when we are in the instruction starting say rather use this platform because of
guidelines for preventing vendor lock-in.
The problem I find is vendor lock-in doesn't happen.
This is how you maintain your job. This is really about like a spark or something. Incurable virus. Classic film.
Azure has another one. So they lock you in at the next level, and that's really hard to get, and it's really hard to actually buy into because there's nothing else really good at it. So Nextflow has its own platform called Secure Platform. Once you go into it,
so it's more.
It's really, I mean, a whole ecosystem. So once you're in it, because there isn't another good.
This is the project manager, I think, you. You come in as a solution.
I know, but like your most that you're using Excel. I mean, sorry, the investment in making this stuff, like capturing the market.
Actively stop to anything that the asking for.
Yeah,
and I feel like
what I mean by
yeah, I agree. They're actively hostile. We should
but one of the things that you can do is facilitate your awareness, right? Or they get swayed by like odd things. You know, so that means that you basically, I mean, what you were saying, but like giving the voice within the technology, because the policy reads like, you know, National Association of Public Health Lab, we have such a
This is a very admirable solution with the national
So all of the rest goes towards someone else that knows how this actually happens.
to get in there for the government.
I agree. Sometimes they won't listen to you. No, they're like, shut up, get in the corner. Nobody listens. But maybe if a bigger body says something.
So because we've got all of these conceptuals, so we are into and then
Yeah, but the thing is that like we need to be like, I mean like literally this is, I mean I was on call with certain networks today, and they said that there was not scientists making themselves available and offering suggestions. We've got this pandemic negotiations. 5 of your things will shut down the lower north. But if you've got 15 suggestions, then that gives you a stronger and they position
What do you think in a situation?
but we stay in our lane.
Captures a lot for this particular point, and maybe we'll move on to the so we have this sort of theme around so sort of having people expanding beyond the competency, so not having enough capacity for individuals, and then underinvestment into the future. So what what mitigations can we have to these risks?
that's a very strange, very specific.
It steals most of our skilled people.
Yes. They'll pay better.
Somehow.
Pay better.
No, but I mean the thing is, so yeah, definitely like pay better thing has some limitations. But yeah, but I mean that so that's like mitigate. So that's why one of the mitigation measures is that I mean I think we have to basically we have to look at like the environment, our environment around
Not an understanding of the workload that's required.
No, I mean, there are pathological cases, like, I mean, there is the head of a African lab that told us can actually see if it's kind of straight, like, they need to stop training. That's pathological. And we can't do much about things like that. But in the best case, where they are, where I think we would do
I don't know anything about economics, so.
So hopefully we're well underway with the mitigations now. I think many of your opportunities could also be mitigations depending on how you're thinking of it.
the
And for these mitigations, let's think, who are the key people that can help? So we've got the blue post-it notes, or your facilitator can write it down.
Yes. A training pathway. Leadership, everything. So some people want to be leadership, some people want to be, I'm thinking like a programmer.
Like a technical kind of.
But when it gets very complicated because like bioinformatics, they think, oh, you can do this, that, so you can do this one, so you can do, like, there's a lot of things I'm struggling with when they're thinking, and I don't work with it. It's very complicated.
So yes, I was saying that like you need to, as you grow, your management needs to expand roles and job descriptions. You know, like this is not going to be, as I said, you know, turn the bench into a web desk. It's not set. And workforce. Yeah. So I mean, and then you, the two, you can't do two. But mentorship keeps coming up, right? And one of the things, so we really are struggling to fund people to come to Phage because like growth funding for public health workers, you can get funding from they're getting money to actually support people in public health labs. You know, to some extent as a scientist, growing to work in public health is where your career dies, right? Because you don't have time to write papers, you know, so like how do you, I mean, so how, because you do have a service job, you cannot be writing a paper on sample need analysis, right? But how do we create a mentorship path in
I would, if I can jump in here, I would really like to see ultimate mentorship where it's like one-on-one instead of you know we do training and it's like one person with 30 people. I really want to see that one-on-one where someone senior takes someone junior, and that could be like someone not just have positions in school, and you could be really good at, say, for example, understand the HIV biology of all the tools that with it, and you can then teach someone the with regards to that particular field. And you can even do it as where you've got two people and they share information. So it's not mentorship, it's information sharing with complementary skills. And I was, because I've been...
I mean, I absolutely agree with you. I'm just not sure. I mean, I've never had a mentor in my life.
Me neither.
So
I would love it.
I mean, I hear about this thing called mentorship. I actually don't know what it is. But the thing is that, like, so what you're describing is opportunities for parents. No, actually do that.
So if you haven't started, now start thinking with the blue post-it notes. Now, who are the people who can help you mitigate these risks or realise these opportunities. So with the blue post-it notes, please.
Yeah, for implementing this mitigation. So can we identify people who? Yeah. I mean, it doesn't have to be individuals, but yeah, like groups or whoever.
No, but yeah, but it is a so I am very interested in the kind of lack of professional societies. Does any of you guys actually have a professional society? We have Public Health Association in South Africa, but within the public health genomic space, I really feel like we're not really ISMB, we're not really.
I think we get like professional society.
Would that be on the opportunity?
That's a big one. There is a professional group within there as well.
It's not very active.
Yeah.
I'm professionally on Slack, and there was this. I've been struggling to connect with one professional, which is I think for us. So let's take some benefits. Benefits, yeah, public health benefits. You know about the benefits Slack, right? Yeah, but he's on benefits. Yeah, that's a good man. That's the best we have. What?
Of all the regions. And then they pay you these, and then they organize meetings.
So don't the field, I mean the field APs have doesn't the field APs have a network like that? African field APs.
Yeah, but that is I think that everything is more, it's not like an organization or like a society, it's more like ASLM or
I mean, there's ASLM, we have African Society of Barge Medicine, but for me, they don't really bring me anything.
yeah, but that's what I mean. Like those are not actual societies. They are
The processing that I work is ISCB, International Society for Microbiology. There's ICB Africa that has an occasional conference, but it's like it's 99% or 90% is research.
so you can have like
But then you can have
country societies and then regional societies, and then you meet. It doesn't even have to be like on Slack, but you connect with each other, so people know each other.
What are those? But I was quite interested earlier hearing the idea of. I mean, maybe this is totally crazy, but I actually thought the idea of like kind of pathogen specific techniques sounded really interesting, like this robot IP thing.
But if you're a bioinformatician, you're working on everything.
Yeah, but I know. I mean, it is a problem, right? I mean,
and there's not a lot.
Yeah. So if we were thinking about who could help with these kind of mitigations, would you say something like that typing or someone would be someone that would help? I mean,
all of this be organized. Yeah. I don't know.
We will help you convene.
But seriously, I do think. I mean, so when I hear you about like the pathogen of the week, but I do think that like the focused disease collaborations give us quite good use cases that can help with certain things. Because if you learn what you learn on typing it, it's not going to stay on something.
No, but you can invite. So we started in South Africa. What was it called? But anyway, so it's like people are doing bioinformatics. It's a small group, and then now we started. So it was South African. All the South Africans and stuff put in, and then we have like twice a month meetings where I would come to the end of Siala, somebody talks about something else, and then we discuss like having.
So the models can be like platform specific. They can be regional. There's different models, but there's basically and I mean like there used to be an ICD specific one that who was it from somebody actually
Yeah, but then they were also quite
Yeah. But I mean the thing is that like there's different opportunities where
society to form, because like everybody, the society that would exist, people want to be okay, I want to get involved in the society, and you have needs in different areas, and then the needs actually are responsible for defining what the society would do and how they would do it.
What's happening in Bangladesh, Malaysia, and things like that in this space? How are you guys developing like skills and workforce and things like that? Do you have societies? How does it work? Yeah, but just within your internet space.
Alright, so we will Tuesday on funding
And so you compete for research grants like our IPSN. But I mean, when you are developing professional skills, I mean, you have a regional, country level or regional data that you But what I mean is, like, where do you find your bioinformatics? Where do they find other bioinformatics?
I have achieved this. I have myself mentored everyone to this level.
And then internationally, you send to conferences. How do they like? Because one of the things you don't want is a lonely bioinformatician, right? That doesn't know who to talk to. So how do you go? Do they join meetings? What do they do?
Two of the colleagues who are supposed to be coming, but they couldn't make it because of
conflicting meetings.
Alright, so basically we do it like this is the reason why Alright, so for us, we do this because we need this. Because otherwise we cannot do it. So my team is nature. So this is what we have achieved over the years by just discharging my mentoring, responsibilities. Also by, how to say, securing funding, continuous funding.
So what you're saying is, what I'm hearing from you related to this is sponsorships to attend. But it's sponsorship for people to attend meetings. So meeting, like there must be, because I mean, I'm doing the same with my research funds. I'm paying for my people to go to conferences. So this is important. Put the so basically travel funding, conference funding. It's really important to not leave your people in just one place.
Yeah, that might be a good one for this travel funding, maybe in the pink.
So we have around 10 more minutes, and then we'll begin feedbacking from each group. So 10 more minutes to capture ideas. Think about what key information you want to feedback from each deliverable. What were the key risks? What are the best mitigation strategies, and who should be involved, and share them with the rest of the group.
So I was attending American Society for Microbiology meeting back in 2024. So there was some kind of inscription displaying that I'm a bioinformatician. Because dearly needed for me. One of my students who returned from China, he was coming to Australia for his postdoc. I stopped him saying that he just stay some time here, then you decide. So now he is a full-fledged bioinformatician. But the support, I have been trained in the US, also in UK. Now he's opportunities also for training.
But the other thing that we need, that is sometimes under-recognized, is that look, people leave. So in other words, better documentation, including SOPs, can ease the fact that people will leave, right? So the too much of what we work with is not well documented. So like, for instance, you know, if you don't, like this is why I promote Galaxy. If you don't need to know command line to do your analysis, that's an advantage when you've got somebody who doesn't need to be 100% Python. You know,
Do you guys have a lack of bioinformaticians that you hire? Or are you using in your country?
This is not a, it's not a traditional post, so something that you have to motivate.
Also, let me share with you my this information I'm talking. It has been so much diligent, so capable. Now the institute, like starting from the top level to influential levels, they are trying to, you know, kind of make the use of him and offering him salary, offering him meeting, you know, support and other things to him, their data analysis.
Yeah. Is that through recognition? So people have somehow, the system has allowed his recognition to sort of come up.
Yeah. No, so I mean, the thing is for the first 20,
yeah.
I mean, the first 15 years of my career.
Who's responsible?
Oh, sorry, this was meant to be paint.
That's all right.
Pink, yes.
I think I mean,
for those that doesn't have the capacity to buy some sort of training, basic bioinformatics, so that at least we know. So when we want to talk to our public health epidemiologists, we need to be sure about the tools that we are using.
Yes.
Not to the until we become experts, but at least we know the necessary.
So so we need not just documentation, but what we actually, the other thing that we actually are seriously missing in our field is textbooks, open textbooks on the methods, right?
Who would be responsible?
So that's I don't know who's responsible, but I mean the thing is that like
All scientists are so doing things for free.
no, I
you
don't.
know, like
I
you write
really
a book
don't
for
know.
free, you write a textbook for free, and then somebody else does it. And so most now you say, can you write this chapter?
Well, I mean, it's a very. Do we fund like
educational funding?
Yeah. So if it's something there's a need for it, like.
So I'm not thinking about this for free. I'm thinking about who would be paid.
I'm sorry, if I can ask, what sort of textbook are we talking about?
So I was looking at. Forget the guy at Cambridge was advertising a course coming up now, £65 a day. It looked excellent, right? Firstly, I was thinking to attend the course just to see what's in its curriculum, pay the money out of the grant, something like that. But the other thing I was thinking is beyond that, the course is going to give you this example, that example, that example. A textbook that can basically say, okay, your techniques for you're going to do QC. This is what QC is about. You're going to do genome assembly. This is what a quantic is. This is what a assembled genome is. This is how we assess it. This is, basically going through the like. That needs to be very, very deep, but basically going through
It's captured here.
No, the documentation is not the same thing.
This is more for like on the job kind of stuff versus the theory sort of. Yeah.
So my background is biology. All our groups ranging multiple disciplines and having the bioinformatics in my group, I have just realized that is one is statistical background, very, very efficient bioinformation. Because I have also sent seriously, it's not biology background students who is this? I mean, learn bioinformatics, but they just turn out to be basically they have limitations. They say that they don't understand many things.
So maybe you can also and what the basic requirements need to be before you get.
Of course, it has to be living, but like, I mean, I don't know.
Academic institutions.
So
yeah, true.
I think it's something maybe at a very high level like WHO can, because they have a lot of
textbooks and
books and things like that. So they can actually make a call to have like a series of textbooks that's available on their website to develop them, and then have people responsible for updating and reviewing.
Yeah, yeah. No, I mean, the thing is, it's going to be like, I don't know if you know the genomic epi handbook. But, I mean, I use that quite often. Editas and Alibaba. And I mean, it's just not the
problem with these textbooks. Few people know about it.
Yeah, but obviously they need to be advertised once they're there. So the other thing that is needed that will help is always updated resource directories. I mean, 100% I'm in favor of fair training materials. Because certain people go to, Mohammed comes to training by me, I learn, but then the materials are not available for somebody else. So fair training materials, I'm very, very passionate about that. Reusable. So this is also what I like the Galaxy stuff. There's so much stuff we've reused from that.
One of the things that Welcome does is that if they train you, they want to see that you also train other people, sometimes do a report. And then if you've done that, they actually start auditing you as a trainer in your region, and they pay you to do it. So it's an incentive for you to actually train.
But you know,
downward.
They are moving towards fair training materials. They didn't have. Well, and I mean, that's good. But like, for instance, EBI has a really excellent intro to command line for and then they got all the materials, and then I realized I relied on the virtual machine image that they hadn't checked. It was too big. So making sure that everything is fully fair, that anybody can pick it up and use it, is really important for training materials. You know? Yeah, but like fair, AIR training materials, right? One of the other issues that is that the boys have to deal with is crossing the language barriers. Because I've really discovered that because we have a huge goal in Africa with Francophone and Anglophone, yeah, depends on who. Angola.
But then, you know, it's if you think about it, all of Africans.
Yeah, but yeah, but Francophone versus, but I mean, but Francophone is a challenge in general, but then of course Arabic as well.
Is that like translation into material translation? Like
so the NTS Academy for us at the University of Cape Town, they're looking into machine translation materials. How more can I do the job? It's not a terrible idea. I don't know what your language situation in your countries is, because it's a I mean, I don't know if, but yeah, so as I said, machine translation is getting better.
Because I think for a long time so it has, like if you go to Belgium,
how many Francophone colleagues have we tried?
No, I know that. So that's why I'm saying the biggest problem is French.
Yeah, because
especially for us.
Because they have French in metropolitan France, they don't speak English. It comes from up there. But because the Francophone world doesn't speak English, many of them.
They do, but they don't like it.
Yeah,
if you are in France, maybe if you speak in English, very few people are
yeah.
But no, but most of the it's sort of different, but a lot of Francophone African countries are moving towards English.
They are all like for
So that is one thing. Another thing in Japan, right, it was very so I've seen the natural journal will translate and immediately appears. So the students now read
I recently discovered that there's a Francophone bioinformatics system that they have you've heard of, that's now developed by French researchers.
Okay, so the groups are coming back in the room together. So in, let's say, three more minutes, we'll begin the feedbacks from each of the groups.
I know, everyone has for the first time. Yes, I think.
All right. Thank you so much for all of the energy and discussion. I see people don't want to stop. There will be all afternoon to continue the discussion over some barbecued goods. So what I'd like to do is ask each of the facilitators to now have the opportunity to feedback and share some of your discussion. So we'll leave it pretty free how the facilitators want to feedback. They can either go through each of their deliverables and talk a little bit about the risk mitigations. I know some people have done some prioritization around risks and are going to feed those back, and that's great as well.
I was thinking with.
So perhaps we can start over in non-numerical order, group 2, equity, access, and capacity. So please share.
Thanks, Tim. Okay, I'll start and then I might hand over to Alicia for anything to add. So equity, access and capacity, I'll go through them objective by objective. So the first objective was mainstream genomics into public health evaluation and training systems. And what the deliverables were including genomic surveillance into existing programs like the JEE, FETP or the GLP, making sure that there's training modules available for mainstreaming genomics into public health, and there's mechanisms for lab system strengthening. And some of the risks that we identified for those are, of course, when you're developing training modules, they need to be current, they need to be they need to consider the scope and also the level of expertise in order to be pitched correctly. In terms of mainstreaming and developing training modules, you also need to consider surveillance system needs and capacity. And there were some questions about if you're adding additional genomics modules into already quite lengthy programs like the GLP or some FETP programs, what does that mean in terms of capacity to actually absorb and build that information into quite an established program? And some of the mitigation strategies, particularly around developing training frameworks, were a lot around communication. So making sure that we're talking to the people that are responsible for the existing programs, making sure that we're communicating and involving the experts that will be able to contribute to the development of the materials, and making sure that we're able to run pilots and assess the feasibility and the suitability of those programs. Do you have anything to add to that one, Alicia?
The only other one that we identified is often in different regions there's different training programs that are being operated by different groups, and it's not clear sometimes if they've got different objectives or if it's sort of muddying the waters a little bit, and whether or not there needs to be some sort of overarching governance and whether or not IPSN could perform that function to coordinate training for genomics across, within different regions and things like that, just to ensure that you've got everything covered and you're not sort of doubling up on your programs.
Great, and I think for that one, in terms of who can help, given it's quite broad in scope, there was everything from ministries of health to donors and funding agencies, training providers and implementing partners, academic institutions, as well as people that have been trained or are responsible for the delivery of training. Okay, so moving on to objective 2, which is overcome procurement and supply bottlenecks, where the deliverables were things like providing and publishing an essential reagents list with use case guidance, being able to undertake a consultation process around that and to disseminate that as a guidance for use. And some of the risks around that was if there is an essential reagents list, what is the backup plan when there's stock out of the reagents on that list? Considerations about the differences in availability of reagents across different regions and different countries, and really thinking about, as an opportunity, how we can take joint consortia or joint purchasing models to really enhance access to reagents. Ooh, anything to add there? And I guess given this is really around supply chains and infrastructure, the who can help was quite big picture. So industry, government, implementing partners, and regional consortia or networks that already exist, particularly for an advocacy perspective. So the third objective was generate local evidence on cost effectiveness and impact. And really this is a recognition of the fact that there's limited evidence or limited implementation research in a lot of settings globally around the use and the utility of genomics. And some of the deliverables were around establishing an implementation research consortium, identifying priority areas for implementation research, developing a position paper, and then doing some advocacy around the position paper and the outputs. And I guess the risks here, and my group focused quite a lot around assessment of cost effectiveness, is how do we access the expertise that's required to do that. So, you need health economists and that kind of expertise, but that's not always accessible to everyone. There are things, so opportunities are there are things like a genomics costing tool and other things in the IPSN toolkit that would be useful. And in order to set priorities, you need to understand the stakeholder priorities. Was there anything else to add from your group?
We just did a bit more. I don't actually think we focused on the cost effectiveness. We really had a look more. We were really thinking more about the impact. And I think the themes that came out of that was to really bring policymakers and public health people along with us to increase the genomic literacy and really leverage existing dashboards and resources that we had to ensure that we've got translation and it's very, very clear what the positive impact can be and whether or not we need to generate a framework or a resource for how we can best demonstrate this in different settings to ensure that the benefit is very, very clear for people that need to make these decisions.
Yeah, I think that's about it. Yeah, and look, I think, in terms of who can help, we pulled out the role of academic institutions here, of implementing partners, of government, of different networks and consortia. But we also spoke about the need to engage media and mainstream communication channels to really be able to communicate the impact of the work. And then the last one is improve representativeness and efficiency in data collection. And a lot of the deliverables here around developing a sampling framework document, a piloted tool around the sampling framework, and then an evaluation report. And some of the risks are that sampling frameworks may need to be context specific. And of course, we've spoken about it a lot over the last few days, but the metadata considerations and how to integrate that into a broad sampling framework. And some of the mitigation strategies were that if it was going to be a sampling framework or a sampling framework guidance at a global level, it should really focus on minimum standards for sampling and with some adaptability and flexibility so it can be tailored to a context-specific setting as required. Anything else to add there?
That was it. And I think the only other thing that our group identified was maybe data collection methods. A lot of them in different, in some settings are static and they're potentially a little bit old-fashioned, or we've been doing them a certain way for a certain period of time, so maybe we need to think about to facilitate data collection depending on the application, and if you're in the field or a different sort of a setting, different tools or methods that people can actually use that would enable them to collect data beyond the conventional ways that we tend to, write things down on a paper list or, store it in an archaic public health database or laboratory database.
Great, thanks Tim.
Great, thanks. Thanks Group 2, just give them a round of applause. Okay, we'll now move over to Group 1.
Group 1.
Over here,
Okay.
so governance, ethics and trust.
Yes, so we had governance, ethics and trust. I'm not going to break things down into each individual objective, although I will read them in a second, just because we found that while our sort of risks do broadly map to the objectives, there was a lot of overlap between some of them, so it was just sort of easier just to group them as sort of major risks together. But the objectives were to support adoption of global genomics guidance into national planning, ensure lawful, ethical and secure data generation and sharing, build a trusted, interoperable global sharing model, and strengthen collaboration and reporting frameworks. So some of the risks that came out were around flexibility, so of the toolkit. So there was a lot of discussion around how do we keep it up to date and have continual improvement. How adaptable to varied context will it be? Do we deal with things like language and cultural barriers? Are there only one ways to do things, or will there be, multiple different ways to achieve the same outcomes? We didn't have a huge number of mitigation factors for this. We've got some more for some of the other ones, but a lot of discussion around learning from other similar processes, in particular sort of away from the pathogen genomic space, that we don't always maybe bring in other industries that are completely unrelated to even health, but that have these same sort of kind of global guidance and sort of continual, need to update those processes and the like. But we did identify that, and also just the importance of really communicating why the toolkit is needed. But the people that were identified as potentially helpful in this scenario: industry, academic partners and funders. When we started to talk about sort of national policies and regulations, we sort of got broken into two different sort of areas. So there was a lot of discussion around issues within countries, so obtaining agreement and participation within countries, and then between countries. So probably by far the most comments were around getting within country agreement and participation. So a lot of people just reiterating how in their settings it's really hard to get consensus around things, getting agreement between things like ministries of health and industries and academia in order to be able to implement national policies. That often it's the same voices heard, and a lot of people that are the most vocal are sort of maybe a little bit resistant to change and a little bit set in the old ways of doing things, and struggling to bridge the gap between implementers and policymakers at a national level. So it's quite a few kind of practical mitigation strategies that people came up with. So making sure that the toolkit includes glossaries of terms so that people that are having these conversations know that they're speaking the same language. Coming up with some of those use cases that show what the value add is of having these national policies and the like. Ensuring that engagement is early and widely and continually with stakeholders. identifying who those interested stakeholders are, and ensuring that any sort of discussions around this have adequate representation, particularly to address some of those issues around it's the same old voices that keep getting heard. There was a comment that the players who are involved in this differ quite a bit between settings, but that civil society organisations, national committees and local champions are important. Between countries, the issues were sort of similar but a little bit different. So there's a lot of discussion around power imbalances. So if we're coming up with sort of things that involve, inter-country sharing and the like, how do we have incentives for smaller players, or how do we address the perceived or real inequality in benefits? I mean, we've already discussed this a lot throughout the meeting. But one of the thoughts that came up was around, with some of these global decision-making processes, bringing in independent mediators and assessors, sort of at intergovernmental or even UN level, engaging ethics experts and, again, national committees. The next sort of, bunch of discussion was around the data platforms and sharing. So there was a comment that there's already a lot of groups trying to do the same thing, and that we need to leverage existing networks and platforms to make sure that we're not introducing more fragmentation, ensuring that there are defined and tiered levels of access and use. And some of the people sort of involved in that might be developers of open source tools, and developers of global platforms, and local developers and bioinformaticians and the like. There's probably more there, but we largely focused on, the technical expertise there. And then there was also quite a number of comments just around the things that would, prevent people from feeling comfortable participating in any sort of global sharing arrangement. So they were things around, how to make data both secure and accessible. How do we prevent people from feeling like they're losing ownership over their data? That there are risks associated with federal models around ineffective communication and decision-making, losing a single point of truth, and who is responsible for sort of interpreting what's actually meant by the data, but also who is responsible for data breaches and loss that comes from any potential data breaches. So there was a lot of discussion around how we really need to engage local points of contact for any global sharing. There need to be a lot of monitoring and evaluation to ensure transparency, continual improvement, and to demonstrate evidence of utility in order to get people to want to participate, and also flexibility on how countries and players interact, so particularly around things like timeliness and completeness of their participation. So we want to maybe introduce things slowly and allow people to start off with only small amounts of participation at first. In terms of the players, there was a lot of discussion around different academic experts that might be useful, so ethics, monitoring and evaluation, and AI experts, that we need to engage policy, legislation, writers and decision makers, but also identifying some of those individual labs or public health agencies and countries that might be early adopters and would be happy to sort of put themselves up as positive use cases. And then the final one was just around incentive models, which we probably had the least discussion on. And that was just around, some of the issues associated with competition and resources, and that we need to generate positive use cases and showcase best practices. And some of the players involve industry, academic publishers, trade regulators, and other regulators. And then the only other discussion we had was not around risks, but actually some of the opportunities that we might be able to leverage to get some of this done. So there was an acknowledgement that there were a couple of external pushes that were happening at the moment that were opportunities to sort of leverage from. So the IHR push for inclusion of genomics, the pandemic agreement, and the AMR independent panel on evidence for action. And then also just an acknowledgement that there are, a number of successful collaborations already and a number of global networks and countries' experiences that everyone can learn from, and that we should really leverage these successful regional networks to sort of provide some of that evidence to deal with some of these risks over here.
Right.
Did I miss anything?
All right, thank you, group one. We'll now go over to group 3, interoperability and standards.
Okay.
Ladies first. All right, so we are Group 3, Interoperability and Standards. So I guess what we'll do is we'll just take it one dimension and synergize. So the first objective was ensure reliability, comparability, and interoperability of national systems. And the deliverable that was going to achieve that was the creation of metadata and data quality policies in a standards repository, and also standardized genomic surveillance external quality assessments, so EQAs. So there were a lot of risks. There was a lot of discussion, but I'm going to try to boil it down until and prioritize just a couple to report. The first was that EQAs require a lot of work, and it's a little bit tricky in terms of understanding who's the authoritative source. So who is the one that should be conducting these EQAs? And so that was a risk. Some of the opportunities or the strategies that we could use are that there are already, I think WHO kind of led models out there, for example, HIV ResNet. And so what could happen is that there probably would need to be an EQA for lots of different pathogens, lots of different programs. The WHO could help in coordinating the creation of a network of different national reference labs. And those different national reference labs could put up their hand and design the EQA, and the WHO could play a role in deployment and coordination. Another type of risk was that there are a lot of different metadata standards that are floating around out there, and sometimes there are policies in place that say a certain nation or a certain institute has to use a particular standard. And so even if we created nice international standards, they wouldn't necessarily be able to be used everywhere. So what would be really important is to create a repository where all those standards can be found, but in addition to that, creating mapping and interchange tools between different standards so that people can use the standards that they have to within their institutions, but as soon as the data needs to go out, it can be translated into different formats and different standards. And Phage is already doing a lot of that work, so we can definitely play a role, and we're already working together with IPSN in creating a standards repository, so that's good. Okay, I'll hand it over to Jake.
Thank you, Emma. Yeah, so enhancing on that, we also discussed some risks like different target user priorities. So depending on the nature of your institute, your public health lab, if you're using, say, AMR results from Curated for patient management, that's a completely different situation to if you're using it for surveillance or research purposes. And there's no real kind of clear fix for that other than making sure you work within scope with the information you have available, and that the data sets and quality policies and stuff like that do actually reflect how you should be using and interpreting that data. The other risk we had different was really aligned with the general theme throughout this event has been the metadata standards. So Phage has been doing some awesome work on kind of fixing all those gaps, but I think we can also learn from kind of the quality policies and metadata from already existing programs like the Global Pneumococcal Sequencing Consortium run by Stephen Bentley. They've actually been able to achieve sequencing for pneumococcus in most countries around the world and produce really high quality standard results. So involving that research arm could be really important for that consulting side.
Another solution was, it kind of is following on from what Jake was saying, is to understand what kinds of metadata are useful in different scenarios and to provide guidance about that, so that not everything necessarily needs to be shared all the time. Yeah. Do you want to do a second?
Yeah, so the second objective that we discussed was to enable insights while protecting sovereignty and privacy. With the deliverable being pilot federated analytical implementations with smart contracts, scalable architecture, use case documentation. And this one, we actually spent quite a bit of time discussing the difference kind of between privacy and sovereignty of the data. So, and we were discussing about how we inherited a lot of our risk on privacy and stuff from like ethical considerations, like in the research programs. And so maybe we need to readdress how we're handling some of those balances and checks. put that forward. And we do already kind of de-identify data in a lot of ways where possible. So maybe there is less of a risk for the privacy aspects for particularly individuals, unless, say, if you're in Victoria and you get salmonella, there's many, many people that get salmonella. So if that data is available, the chances of identifying you, even with like year, month, day information, is pretty low. But if you contract an extremely rare disease, then you're probably much more identifiable if people know that you've got that disease and live in this certain area.
So we felt, in our discussions, we felt that some of the biggest risks of developing federated systems is that they're very inherently complex, and they take a lot of work and a lot of technical expertise. And it was sort of felt that, I mean, they're would be lovely to have, but they may be overkill in some situations, and so a federated system is maybe not necessary in all situations. And so what would be good would be to explore alternatives to federation as well, and sort of build a catalog of different sort of infrastructure possibilities, catalog the different success stories and sort of adaptive strategies that go along with those systems. So something might work really well in one place. It can't necessarily be exactly replicated somewhere else, but it could be adapted and be made successful in different places. And I think the phage infrastructure working group is working on something along those lines right now. I know. Sorry. Do we want to move on to the next one, or do you want to? Okay, all right. So the third objective would be to help to build literacy on legal and policy aspects of genomic data use, reuse, and sharing. And that would be done through the development of white paper, draft standards, benefit sharing frameworks, and policymaker training. So in our conversations, what we were talking about was the risk is that their policymakers are still a little bit unaware of the benefits of genomics, but also the negative impacts of not sharing genomics as well. So what we were thinking we could do is compile not only success stories. Everybody talks about compiling success stories, but also compiling disaster stories. So having a little bit of a shock factor to counterbalance, the solutions from the success stories. And I'm sure WHO, and there are lots of other groups that could probably help with, and GMI could help catalog those stories. And then another issue is that there could be lack of consensus on best practices, especially for standards. And so the WHO and GMI could convene a work group to get different standards groups together to hopefully hammer out those differences.
Thank you. Yeah, and building on from that, we were discussing that this aim's actually quite complex because of all the required domain knowledge. It's not just knowing what's needed in your country and the setting, but also all the legal requirements, and that's really, I think, where it merges with the Group 1 governance aims as well, which becomes really challenging, because how do you get lawyers to understand all the intricacies of genomics and all that? So it's only really through kind of a long-term strategy and working groups that you're going to be able to get to that common consensus of language, so that you can then do all the country-specific implementations that you need to do. All right, so we'll jump on to our fourth objective, which is build trust and reduce barriers by validating tools against public health criteria. And really, with the deliverable being a tool endorsement process, benchmarking platform, and a repository of endorsed tools. And so kind of the first question that came to mind is, who would host this valuable resource? Because I think Ebenezer said it quite nicely earlier, that if stuff is out of country, that becomes a bit of a challenge. So do you instead have a multi-jurisdictional setup from a singular entity that then is mirrored in all the different countries, where people can host their own versions of the tools that they need to use? And because then that comes into account with the volume of data that you're using. Are you doing things and in high-performance computing cluster, analysing thousands of samples at a time, or are you only doing smaller scales? Those needs You need the system to be able to work on a laptop, and you need it to work on a high-performance cluster. I think we can learn a lot. So some key players, I think, in this area are people like Keith Jolly, right, who have done amazing work for such a long time with PubMLST. They've seen the changes in genomics over the years and all the different requirements. Like for those of you that use that now, you now need to have a login to download that data now, and that's because they weren't keeping track of their metrics enough to keep ongoing funding models. So there's clearly a lot of intricacies about hosting these platforms, and I think we can learn from people who have done it the hard way, and the Pasteur Institute is another example of that.
So we had a lot of the similar discussions. One additional risk was this feeling like if there is a list of endorsed tools, that may make labs feel a little bit stifled in terms of building new tools or finding appropriate tools for their needs. And so really having a checklist of criteria, what is that criteria, first of all, and who's going to create that checklist would be really key, so that as you're building new tools that are specific to your use, you could be validating your particular tool against the checklist. You don't necessarily have to have it accredited by a third party. And not to sound like, not to keep repeating myself, but Phage has been doing, in the Pipelines and Visualizations Working Group, they've been creating some guidance around this, so that could be a good place to start. And then another risk was the sustainability and maintenance of tools over time. So, you can be building or you can be accrediting tools, but if they're not maintained, then that accreditation is a bit useless. So we are hoping that funders will start to recognize the importance of maintenance and be funding maintenance and to help sustain tools over time and not just always chase innovation.
All right,
and one last thing just to add, I think Emma pointed out really nicely about the differences in tools. So with the ring trial and EQA that we're putting together, Melbourne and Zurich, our QC pipelines only really have like 2 or 3 tools that are the same out of like 20 or 30 tools in each pipeline, yet we're able to get a 99% concordance in the results coming through. So I think that shows, rather than certain tools locking down, having an equivalency process would be extremely important to deal with people's different capacities. So.
All right, thanks, Group 3. And then last but not least, sustainability and system resilience.
All right, we will be quick because we distilled down our discussions to a few points. So the first part, the first objective was to demonstrate long-term value of genomics investments, deliverables being investment cases, cost-benefit analysis, and published case studies. So we talked a lot about the reuse of data, and we saw that one of the risks were that the data was not being appropriately reused to the extent that it could be. That also included actually infrastructure, so you don't let sequences just sit and not being used when it could be used for other things. Some of the mitigation strategies was applying the FAIR principles to both the data and the infrastructure. And unfortunately, I think that who could help with this comes down to having stable long-term funding from diverse sources. So we spoke a little bit about sort of what happens now when you have the USAID not being able to fund a lot of the work that it's doing, and the vulnerability that such a thing creates, so that we need to be aware of that we constantly had multiple funding sources. The second objective was reduce duplication, align donor and national priorities. Deliverables being funding landscape mapping, agreement templates, and priority setting reports. We discussed a lot around the priorities here. And the major risk was the discussion around that one size does not fit all in relation to priorities, where you would have a lot of top-down things. So the WHO putting out principles of this is the priority pathogens that we should be looking at. One of our colleagues from the Africa CDC said, well, you have from the Africa CDC lists of priority pathogens, but that does not always fit all of the nations. So some of the mitigation strategies there would be around fostering collaboration between the different layers, both geographically, different nations or within nations, but also between different principles. So you will have patients all the way up to the public health professionals that deliver this. And the help really was around having a bottom-up strategy. So you have these meetings where we discuss these things, but also to include that we have everybody from different nations, but also from different parts of the public health column. And don't read into that we don't sit close to each other. It just happened to be that we discussed the first two mostly, and they discussed the second two.
Thanks. Yeah, so we had the same group, sustainability and system resilience. And in our, we focused on two of the points, to build sustainable skilled networks and leadership. Was the first one, and the deliverables were a competency framework, mentoring programs, and leadership initiatives. And I'll just go to the sort of focus of our discussion, which was on the expansion of tasks beyond the competencies, beyond the capacity of a role. So if, as we're in a very dynamic field, things are changing a lot, and that you can often end up doing a lot more work than you initially signed up for. And the second sort of major risk we identified was an underinvestment in future leaders. And we came up with a lot of different mitigations. So this included defined career pathways based on competencies, enabling travel funding for people to be able to travel and meet and expand their network, ensuring that roles are expanded with the changes in the tasks that you have to do. And then, yeah, training pathways, so having pathways that show how we can increase in our training over time, as well as we've also spoke about the FAIR principles around training materials, and possibly having more textbooks, because a lot of bioinformatics in particular doesn't have a lot of textbooks available. And yeah, there was some discussion around how this would be paid for, or where funding would come from this. An increase in professional societies and networks. So again, enabling sort of leadership through these inter-institute societies across countries. And yeah, for the sort of expansion, we focused a little bit on training materials as well. So having resources available through a resource database, doing translation of these materials into different languages. So if the materials aren't available in your language, that can sort of stifle your skill advancement. So having that done through AI or some sort of machine translation methods. And then also increasing documentation so that the jobs have clear documentation for how they're supposed to be performed. And then another important one was recognition of people's role, so that in a organization, people at the top are able to see the work that you're doing through some sort of formalized recognition. The people who there's pretty broad of like who would be helpful in this for these tasks, and this include professional societies, World Health Organization, IPSN, CDC, Galaxy, who does a lot of open source training, and then academic institutes. In our second point, we focused on ensuring resilience for bioinformatics infrastructure, and this was sort of cloud cluster frameworks, API, workflow repositories, QC pipelines, and governance mechanisms. And the main sort of risk we focused on was cloud platform lock-in, and so that's sort of the idea that you develop your pipelines for a single infrastructure situation, and then you're sort of trapped in that Azure cloud or AWS cloud or something like this. And so the sort of mitigations we were thinking about was investing in cross-platform capable infrastructure components, possibly pooling our procurement to mitigate lock-in. So as a whole, sort of as a community sort of coming together to define guidelines for how we might prevent lock-in, or basically come together to have some sway on these cloud platforms, and then developing some technical policies or briefs to raise awareness of the risk of these potential lock-ins. I might just end there for time. Thanks.
Okay, so thank you, group 4. So that brings an end to the workshop. Just a really heartfelt thank you for all of your enthusiasm and great points there are going to be captured. So this roadmap is going to be further iterated. The Global Partners Forum Phage Conference in October, and there'll be some other opportunities to engage with this journey. But really appreciate your valuable input, and thanks again to the facilitators as well for providing all of that facilitation as well. So thanks again for everyone.
Okay, I'm not going to do an official closing because we have tomorrow to do that. But thank you so much, everyone, for another fantastic day. We are now going to move to lunch and our animal wildlife encounter experience. Now, there's 100 of us, so please stick together because we have a little bit of a ways to go. Everyone needs to pack up their things because we're going to leave from there, more or less. You can leave it here if you want to, but if you come back, I can't trust that anybody's going to be here when you do. So please pack up your things. We're going to head down to the university. Hassini in the back in a pink scarf is going to lead the group together with Torsten, who is very tall, so you can't miss him. There will be a few of us shuffling from behind. Do your best to stay together, and we'll meet there, right in front of the Welcome Wominjeka sign. It is huge. You can't miss it, and it's blue. So if you stand there, we'll take our official photo. Okay, that was loud enough, I think.